Hi, welcome to my presentation on nervous tissue, and we will also cover some basics on the nervous system. So first we'll start by looking at how the nervous system is organized. So in the nervous system, you have two basic parts of it. First, the central nervous system, which is the brain and the spinal cord, and then the peripheral nervous system, which is all of the nerves that come out of the central nervous system or go back into it. So basically, the nerves. So the central nervous system's main job is to process information. The peripheral nervous system's main job is to take sensory information into the central nervous system, so it can be processed, and to carry commands from the central nervous system out to the body. If you look at the peripheral nervous system, we can divide it in a couple of different ways. So first is divided into the sensory division and the motor division. The sensory division is also called the afferent division because it's going into the nervous system. The motor division is also called the efferent division because it's coming out from the central nervous system. So the a there in afferent means to, and the e in efferent means from, like exit. So the sensory division can also be subdivided into the somatic sensory division and the visceral sensory division. So the somatic sensory division is carrying sensory information into the brain that you are aware of. So that would be like, touch, this coming from the skin, visual information from the eyes, auditory information from the ears. Any type of sensory information that you're consciously aware of is going to fall into the somatic sensory division. Then you also have the visceral sensory division, which is all of the sensory information that you are usually not aware of. So that would be like mostly stretch in your internal organs. Also can be some chemical information. So in certain parts of your body you have chemoreceptors that are sensing the chemistry of your blood. So that's part of the visceral sensory division as well. But for the most part it would be stretched. Like for instance, if your stomach is stretched or your bladder is stretched. And then if that stretch becomes really extreme, then you may notice that you're feeling some pain. So you might become aware of it if it's extreme. But in general, you're not aware of that information. Then in the motor division, you can also subdivide that into the somatic motor division and the visceral motor division, but we also call the visceral motor division the autonomic nervous system. So the somatic motor division is all of the motor information that you are kind of voluntarily controlling. So basically, that would be commands that are going to your skeletal muscle, which is the type of muscle that you're voluntarily or consciously able to move. The visceral motor division, or the autonomic nervous system, is all of the motor information that you are not able to control consciously. So that would include like commands going to the heart muscle to speed it up or slow it down, or commands going to smooth muscle in different parts of your body, maybe smooth muscle that lines your blood vessels, so causing those blood vessels to relax or to contract and therefore controlling your blood pressure. So all that stuff, that's all motor stuff that's going on, but you're not able to control any of it directly. So that's all visceral and is part of the autonomic nervous system. The final thing that is in the autonomic nervous system would be commands that are going out to glands, like glands that are releasing hormones in your body or some other substance. So we can kind of categorize the neurons that are in these parts of the nervous system. In the peripheral nervous system, you have sensory neurons in the sensory division and motor neurons in the motor division. Sensory neurons are going to, be picking up sensory information from the world. So they would have either sensory receptors themselves, so they can directly sense information, or they would be in contact with specialized sensory cells that you would have in, for instance, your ears that are picking up information and then sending it to those sensory neurons. Then motor neurons are going to be any neuron in the motor division of the peripheral nervous system. So they're all going to be contacting muscles or glands and controlling that muscle or controlling that gland. Then in the central nervous system, you have interneurons. Interneurons are only connected to other neurons. So they get messages from other neurons and they send messages to other neurons. And their basic job is to process information. So now we'll start looking at nervous tissue and the basic characteristics and cell types that you have in nervous tissue. So you only find nervous tissue in the nervous system. It's not found in any other organ system in the body. You have a lot of cells in nervous tissue, but you also have a lot of extracellular matrix. So in epithelial tissue, it's basically all cells, no ECM. In connective tissue, it's basically all ECM and very little cells. In nervous tissue, it's more like a balance between cells and ECM. In the extracellular matrix of nervous tissue, you have mostly ground substance. You have very, very few protein fibers there. Because you have so few protein fibers, it ends up being very weak tissue. In epithelial tissue, you also don't have a lot of protein fibers, of course, because you just have like very little ECM at all. But all those cells in there are going to be attached to each other using desmosomes, which gives it strength. But in the nervous system or in nervous tissue, the cells are kind of like not really attached to each other. They're kind of just floating in the ECM. So you don't have desmosomes between them, so you can't get strength from there. And you don't have protein fibers really, so you can't get strength from there. And that leaves you with just like nothing for strength. So it's very, very weak tissue, very fragile, very easy to damage. And that's the reason why it is encased in bone in the skull or in the vertebral column, to keep it safe. The types of cells you have in nervous tissue can be divided into either neurons or glia. Neurons are the cells that are actually sending messages in their nervous system and processing information. Glia are also called neuroglia, and they have support roles. You've got 6 types of glial cells in different parts of the nervous system. You have astrocytes, microglia, oligodendrocytes, ependymal cells, Schwann cells, and cytolite cells. And we'll go through each of those cell types in turn. And starting with neurons themselves, actually. So we'll go over the structure of neurons and then go through the glial cells. So all neurons have kind of basic parts of their structure that are the same. They actually have a lot of diversity in terms of their shape, but they always have certain parts. First, of course, you're going to have a cell body. The cell body is a little different from most cells in terms of what organelles it has. It's going to have a bunch of ribosomes and a bunch of rough endoplasmic reticulum and a large Golgi apparatus. So those 3 things, you need them to make and sort proteins. Ribosomes, of course, make proteins. The rough endoplasmic reticulum is going to be kind of folding proteins up and modifying them, and then it will send them out to the Golgi apparatus. And the Golgi apparatus is going to further modify those proteins and then just sort them based on what destination they have. And it'll make sure that they get to their correct destination. So in neurons, you have a lot of different kind of parts of the cell, and different parts need to have specific proteins in them if they're going to function properly. So the Golgi apparatus is really important in neurons. And then finally, you have a lot of mitochondria because neurons use a lot of energy, so they need to be able to make a lot of ATP. Apart from the cell body, you have processes coming off of the cell body, and you've got 2 different types of them. You have dendrites coming off much of the cell body, and then you have one axon. So every neuron is going to have just one axon. It could have multiple dendrites. Some of them would just have one dendrite, actually. Some of them maybe just a couple. Some of them have like a crap load. So some of them have, well, I guess there's just a lot of variety in how many dendrites the neurons can have. They can also be branched, like a lot or a little, and then they're all going to have just one axon. The dendrites are always used for receiving messages, and the axon is always used to send messages. So here you have a basic diagram of a neuron. You have the cell body in the middle here, and then in yellow you've got dendrites coming off of it. So this neuron has a lot of different dendrites, and you can see that they're branched quite a bit, although there's other neurons that have even more branches coming off of their dendrites than what's shown here. It can get really crazy. Some of them look kind of like, basically like trees. So all these dendrites would be receiving messages from other neurons. Then the messages are going to travel to the axon, the beginning of the axon, and if the messages are kind of strong enough, then it's going to cause the axon to send a message of its own. And that message will go maybe to another neuron, but in this case, this axon is shown traveling down to some target cells here, which would be cells in a gland. So it's actually going to be a motor neuron that is telling these glands to release whatever substance they produce. So this neuron is receiving messages on its dendrites and then sending a message along its axon to these gland cells to make them release, whatever they produce. You can also see this axon has a branch in it right here that comes off. So when the axon sends a message, it's going to travel down this branch also, and that might be connected to some other cells, possibly or probably actually some other gland cells like this. So axons can be branched as well, or they cannot be branched. It just depends. But there's always going to be just the one axon, and it'll always send messages. Then you have a kind of robust or well-developed cytoskeleton in neurons that is kind of connecting the cell body to the processes. So the cytoskeleton, of course, is the protein framework that is, giving the cell structure and organization. And of course, the dendrites and the axon can have a very complicated shape, and that shape is going to be maintained by the cytoskeleton. And it also is necessary for transporting proteins from one part of the cell to the other. So if you have a protein that's coming out from the cell body and it needs to go to the end of the axon, it's going to have to travel down there following the cytoskeleton of that axon. So we'll look at those processes in a little bit more detail. First for the dendrites. So we said that they're receiving messages. Most of the time, those messages would be coming from another neuron that the neuron in question is attached to. So you would have probably another neuron off here, which is not really shown, but it would be sending its axon to the dendrites of this neuron and sending a message to them. So when these dendrites are receiving a message, usually that's a message that was sent to them by another neuron using its axon. And the message is actually going to be in the form of a chemical called a neurotransmitter. So this other neuron is going to release a neurotransmitter from its axon that will go to this dendrite and bind it and kind of activate it. Once the dendrite receives the neurotransmitter's signal, it's going to start having an electrical current travel along it. So when the dendrite is carrying its message from the tip of the dendrite into the cell body, that's actually an electrical current that is traveling along the dendrite into the cell body. You may have heard that neurons use messages that are called nerve impulses or action potentials, but that's not the type of message that is going to be found in a dendrite, or that's not the type of current or electrical signal that is found in a dendrite. So that's a pretty large current in a nerve impulse or action potential, but in dendrites you're going to have small currents that are called local potentials or else graded potentials. So you're going to have local potentials starting in these dendrites and coming into the cell body. And they're going to actually end up traveling across the cell body in towards the beginning of the axon. And at the very base of the axon, you have a structure called the axon hillock. When the local potentials get to the axon hillock, they're all going to kind of add together. And if they're big enough, when they're all added together, then that is going to start an action potential. And the action potential is that really large, strong electrical signal that is going to travel down the axon and carry a message to another cell. In this case, it would be carrying a message to these glands to start secreting something. So as we just mentioned, axons carry messages that are called action potentials. The action potential is a really large or strong electrical current that's going to travel along the entire axon. It'll also travel down any branches that the axon has. And as it's traveling, it doesn't lose strength. So if you're receiving neurotransmitters on these dendrites and that's causing local potentials, and those local potentials are traveling towards the cell body and then across the cell body, they're going to get smaller as they go. So they start, kind of bigger, and they get smaller and smaller and smaller as they travel. With an action potential, it's not that way. It's going to start at the axon hillock if the local potentials add up there to be big enough to trigger it, and then it's going to travel down the whole axon and any branches that the axon has, and it's not going to lose any strength at all as it travels. So it'll be the same, exactly the same the whole way, the whole length of the axon. And because of that, we say that action potentials are all or nothing. You either have one or you don't have one. They're always the same, and you either have one or not. Then you can see that this axon here is shown with these little blue kind of bands wrapped around it. So this blue stuff here is myelin. Axons are frequently what we call myelinated, which means just that they have these blue bands around them. And this myelin here that's forming these bands is made up out of a really fatty substance. The purpose of it is just to serve as electrical insulation, basically, and it makes action potentials travel faster. So when an axon is covered with these myelin sheaths and is myelinated, then it is insulated and it'll travel. Well, it'll send action potentials a lot faster, like a lot faster. So not every axon is myelinated, but a lot of them are, and any axon that is going to be traveling over a large distance tends to be myelinated. There's like a couple exceptions to that, but for the most part, every axon that is sending a message over a large distance is going to be myelinated. That way the message can get there without like a long time passing by. Then we mentioned that axons can branch. Those branches are going to have kind of special names. So this big branch is coming out of the axon here that's also myelinated. That is called an axon collateral. Then at the end of every branch of the axon, you're going to have a bunch of these tiny little branches that form. These little branches are called telodendria. Each of these little branches at its end is going to have what you call an axon terminal, which is the point where it connects with the cell that it is sending its message to. And that connection is called a synapse. So we'll look at a synapse and the structure of a synapse right now. But before we get into the synapse, I wanted to show this example of an axon. So what you're seeing here, all these little lines, are the branches of a single axon that were mapped out and drawn onto this map. This axon is found in the olfactory cortex of a guinea pig, which is the part of the guinea pig's brain that is processing smells. So over here you have kind of a top-down view. This is like the front half of a guinea pig brain. And here you have the olfactory cortex, and right here is the cell body for this particular neuron. All these lines coming out of it are the branches of its axon. So that is literally one axon that is branching like a ridiculous amount of times, and those branches are traveling pretty far in the brain away from this cell body in both directions. Over here you have more of a frontal view of the guinea pig brain, and there's actually multiple slices here that are kind of superimposed on each other. So here you actually have one frontal slice. This is another frontal slice. This is another one, and then this is the last one. So it's just like if you were facing the guinea pig head on and you were slicing its brain up. This one would be, the slice that is most in front, and then second most in front, and then, behind that one, and the slice in the back. So we're watching, Okay, so we're basically watching the axon travel backwards in the guinea pig brain and forwards in the guinea pig brain. So just another view of those axons. Basically the point is, or I should say the branches of that single axon. The point of it is just that, axons can be really branched and have a very complicated structure. It's actually estimated that this particular axon was making 8,000 synapses, which would mean that it has 8,000 connections to other neurons and is sending messages to, to 8,000, potentially 8,000 other neurons, unless it's contacting the same neuron multiple times, which it actually might do. So actually, one more thing before we talk about a synapse, which is axonal transport, which is just, how the neuron is transporting stuff along its axon. So if you're looking at an axon at the internal, the interior of an axon, there's really not much there except for the cytoplasm and the cytoskeleton. And we have a special name for the cytoplasm that is inside of an axon. We call it the axoplasm. So it's really just, full of the cytoskeleton and then with some cytoplasm around it. You really have little of anything else in there at all. So you're not really going to have organelles in the axon. But when you get to the end of the axon, at the axon terminals, then you actually have like, this kind of knob that widens out a little bit. You don't have as much cytoskeleton in there, and you're going to have some mitochondria. Actually, not just some mitochondria, more like a lot of mitochondria, because you're always sending messages from the axon terminal across to another neuron or to a gland or something, and it costs energy to send those messages. So then well, I guess I should say the neuron is using a lot of energy at its axon end specifically, and so it needs a lot of mitochondria there. But of course, those mitochondria would actually be formed in the main cell body. Also, all proteins are going to be formed in the cell body. So any proteins that you need at the end of the axon and any mitochondria that you need at the end of the axon, they're going to be made in the main cell body, then they need to be shipped out along the axon using the cytoskeleton. And that's called axonal transport. So you have a few types of axonal transport. You can categorize it as either fast or slow. If you're doing the fast axonal transport, that always is going to cost ATP, and you're going to have an actual motor protein kind of walking along the cytoskeleton carrying something. Maybe an organelle or maybe a vesicle containing some proteins. So this is actually an image of that. Here you have a protein in the cytoskeleton, so it would be maybe one of these proteins that are shown as part of the cytoskeleton in this axon. Then you have in blue some motor proteins that are walking along that cytoskeleton protein using ATP. And then in purple you have what they're carrying, which in this case is an organelle, so it's probably a mitochondrion. So that's the fast axonal transport. Then you also have slow axonal transport, which of course, goes slow, and it doesn't involve using ATP and motor proteins to move stuff. It actually just involves free proteins that are being sent. So they're not even going to be packaged into a vesicle. They'll just be like freely floating in the cytoplasm. Then you have anterograde versus retrograde axonal transport. Anterograde means forward and retrograde means backward. So just whether you're sending stuff from the cell body out to the end of the axon or whether you're sending stuff from the end of the axon back into the cell body, which you would do whenever stuff gets worn out down there and you need to recycle it. Things can only be recycled in the main cell body, so you got to send them back. If you're sending stuff slowly, so if you're sending free proteins, that's always going to be anterograde axonal transport. So if you're sending something retrograde, so back to the main cell body, you're always sending it quickly in a way that uses ATP. Then I guess this diagram here is also just showing stuff being sent by anterograde axonal transport versus retrograde. So in this case, we would actually be sending some proteins out to the end of the axon by anterograde transport. So we would make those proteins in the main cell body. Of course, you have the genes in the nucleus, so you're going to make mRNA in the nucleus for that particular protein. Then it's going to be sent out to the rough endoplasmic reticulum, which is rough because it's studded with ribosomes. So all those little dots there are ribosomes. So the ribosome is going to use the mRNA to build a protein. That protein will fold up inside of the rough endoplasmic reticulum and be modified, and then it'll be sent out in a vesicle to the Golgi apparatus, which is going to modify it some more and make sure it gets sorted to the proper place, which in this case is going down the axon. So the Golgi apparatus is actually going to package that protein into a vesicle, which we see coming out here. So this would be traveling by fast anterograde transport. You would actually have motor proteins walking it along the cytoskeleton until it gets to the end of the axon at the axon terminal, and then it's going to be used. On the other hand, over here, you have some worn out components of the axon terminal that are being shipped back by retrograde transport to the main cell body for recycling. And this is just showing that same thing, basically, the anterograde transport versus the retrograde transport, but it's just showing, I guess, the motor proteins on the actual cytoskeleton carrying stuff down to the end of the axon or back to the cell body. In this case, you have an actual mitochondrion that's traveling back to the cell body so that it can be recycled because it's probably worn out. And now for the parts of a synapse. So again, a synapse is a place where neurons are connecting to each other. So you're going to have the axon terminal of one neuron connecting to the dendrite of another neuron. So that's shown down here. Here you have your first neuron and its axon terminals at the end of its telodendria here. And one of these axon terminals is forming a synapse with the dendrite of the second neuron. So we're going to have special names for those two neurons. The first neuron is the presynaptic neuron. It's going to be sending a message on its action potential that's going to arrive at the synapse. Then the second neuron is the postsynaptic neuron, and it's going to be receiving that message from the presynaptic neuron on its dendrites. Then if you zoom in on the synapse, you would see that actually the axon terminal for the presynaptic neuron doesn't quite touch the dendrite for the postsynaptic neuron. There's actually a tiny space between them, and that's called the synaptic cleft. So here you have kind of a zoomed in view of a synapse. This is the axon terminal for your presynaptic neuron. Here you have your dendrite for the postsynaptic neuron, and you have this little space between them. That's the synaptic cleft. And you can see some little dots that are crossing this space. Those are neurotransmitters. So whenever you have an action potential come down to the end of this axon and get to the axon terminal, that's going to cause neurotransmitters to be released into the synaptic cleft. Those neurotransmitters are going to cross the synaptic cleft and deliver the message to the postsynaptic neuron on its dendrite. When the neurotransmitters are actually delivering that message, what they're going to do is bind to neurotransmitter receptors, which are proteins that are found on the membrane of the dendrite, right where it's like, facing the synaptic cleft. So for every neurotransmitter, you're going to have one or more neurotransmitter receptors that can receive the message from that transmitter. When the neurotransmitter binds to its neurotransmitter receptor, then you're going to have something happen. What happens exactly is going to depend on what type of receptor this is, but something is going to happen in the dendrite of the cell that leads to the message being sent on the dendrite. Then after you have these neurotransmitters being released, they're actually going to be pumped back into the presynaptic neuron, into its axon terminal. So actually, when you're looking at this axon terminal, before the neurotransmitters are released, they're stored in vesicles, which you can see here. When they're being released, these vesicles are actually going to come up to the membrane of the axon terminal and kind of fuse with it. So this vesicle here is like in the process of fusing, and here you have another vesicle that already fused. When that happens, the neurotransmitters are released into the synaptic cleft. They cross it, they bind the receptor and send their message, and then they're going to let go of the receptor and come back to the presynaptic side of the synaptic cleft, and there they're going to find a neurotransmitter transporter, which is a pump that is going to take that neurotransmitter in the synaptic cleft and pump it back into the presynaptic cell, where it can be packaged back into a vesicle and be reused later. So neurotransmitter receptors are receiving messages sent by neurotransmitters, and you find them on the dendrite. Neurotransmitter transporters are recycling neurotransmitters by pumping them back into the presynaptic neuron, and you're going to find them on of course, the axon terminal of that presynaptic neuron. And I'll just mention that a lot of the drugs we have that interact with the brain are going to be affecting neurotransmitter receptors or neurotransmitter transporters. So for instance, a lot of antidepressants are SSRIs. That stands for selective serotonin reuptake inhibitor, which means basically that those drugs are going to bind to a transporter for serotonin and prevent it from being recycled. So that whenever you do release some serotonin, it's going to end up just hanging out in the synaptic cleft, and for some people that makes them feel better, having more serotonin. On the other hand, psychedelics like LSD will actually bind to the certain receptors for serotonin and activate those receptors. So it looks like a serotonin signal is being sent even if serotonin isn't released. Okay, now we'll go on to the glial cells and the types of glial cells. But first, just a few general comments on the glial cells. There's a lot more glial cells than there are neurons. In fact, there's about 10 times more glial cells in the nervous system than you have neurons. And they're also a lot smaller than neurons. Neurons are kind of really big cells. And also they can divide. So neurons actually do not divide under normal circumstances. Just, you kind of have them and, they are supposed to live your whole life and they will not divide. Actually, in some cases, you maybe can get more of them, but not because the neurons themselves are dividing. It would actually be because you have stem cells in your bone marrow that are kind of, dividing to become neurons and then traveling in the blood to the brain. But neurons themselves do not divide. Glial cells, on the other hand, do divide, which means that anytime you have a brain tumor, it's much more likely to be made from glial cells than to be made from neurons. So it's really hard to have mutations occur in a neuron that make it divide, but since glial cells divide anyways, it's kind of a lot easier to make them just kind of lose control over their cell division, so they divide really quick and then you get a tumor. In the central nervous system, so the brain and the spinal cord, you have 4 different types of glial cells, and in the peripheral nervous system, you have 2 types of glial cells. So we'll go through those in turn. So in the central nervous system, the first type of glial cell that you have is the astrocytes. These have a lot of different functions, and they're called astrocytes because they kind of look like stars. So cyte there means cell, and astro means star or star-like. So you can see those cells here in green. They have a lot of processes coming out from this main cell body in all directions, and that kind of makes them look like a star. So they're called astrocytes. One of the things that they do is help provide kind of structural support in the brain. They can actually kind of hold neurons in place, and they also will send out processes to capillaries and kind of wrap little feet around the capillaries, which also kind of helps form the blood-brain barrier, which is just the barrier between the blood and the brain, that doesn't allow stuff that's in the blood to get into the brain unless you actually need it. Another job of astrocytes is to recycle neurotransmitters and other substances that come from synapses. So I had just, kind of talked about how when neurotransmitters are released, they're going to be recycled in a neurotransmitter transporter and taken back up into the neuron that released them. That's actually not the only thing that can happen though. So some neurotransmitters would be taken up by neurotransmitter transporters into the presynaptic neuron that released them, but others would actually be taken up into an astrocyte, also by a neurotransmitter transporter. But in this case, it's on an astrocyte. So in that sense, they're kind of important for the functioning of synapses and kind of keeping synapses healthy. Also, I had mentioned they have those infeet that wrap around capillaries, and that helps you form the blood-brain barrier. So the blood-brain barrier is really important because you have a lot of things in the blood that might be bad for the brain if they got inside of it. So for instance, you might have something toxic in your blood, and it's really important to keep that out of the brain because the brain is like, controlling your whole body. So it's really bad if things go wrong with the brain. Also, a lot of hormones that you have in your body actually serve as neurotransmitters in the brain. So like dopamine and serotonin, those are neurotransmitters in the brain, and we're used to thinking about them as neurotransmitters. But they're also released in different parts of your body as hormones. So you're going to have some dopamine and some serotonin in your blood a lot of the time, and it's acting as a hormone there. But it's really important that those hormones don't get into the brain because these signals that are sent in the brain need to be related to the actual, function of the brain. If you have just hormones leaking into the brain and then ending up sending signals in the brain, that's going to mean like basically chaos. The function of the brain is going to break down if it's not able to know like that when a signal arrives, it was sent intentionally by another neuron. If it was just kind of like a random hormone coming by, that's basically going to be like being on drugs constantly, and it won't be good. So the blood-brain barrier is protecting you from all of that, and you have to have the astrocytes to form it in the first place. Also, if you have an injury that occurs to the brain, you're going to form a special type of scar called a glial scar, which is actually made by the astrocytes. which I guess we kind of already went over in histology, but yeah, so it is the astrocytes that you need to form that glial scar. And once you form the scar, the benefit of it is that you maintain the blood-brain barrier, but the drawback is that it's kind of this no-go zone where axons can't cross it, so it makes it harder to reestablish any connections that were broken, because they're going to kind of have to try to find their way around it if they want to rebuild, and that's really hard to do. Another function of astrocytes is to kind of influence the way that neurons signal to each other, and to influence the way that the brain changes over time. So the brain changing over time is called plasticity. And it's kind of like the way that neurons are connected to each other. If that changes, that's plasticity. Also, if the way that neurons function changes, that's plasticity as well. So plasticity is really, really important for learning and memory. Anytime you're learning something, that means that the structure or function of your brain is changing. In order to learn the thing. So you really need astrocytes in order to kind of do that and do it properly. And also astrocytes are participating in signaling between neurons. So we have this concept now of the tripartite synapse, and we used to think that a synapse is just between like the presynaptic neuron and the postsynaptic neuron. But now we know that in a lot of those synapses, you actually have an axon with its process like real nearby, like basically right there up at the synaptic cleft. And it is participating in signaling as well. So it can release its own transmitters that send messages to the presynaptic neuron or to the postsynaptic neuron. And yeah, so astrocytes apparently have a role in signaling as well. They're kind of multifunctional and really, really important. So here you have just a basic diagram of that tripartite synapse. Here you have your presynaptic neuron releasing neurotransmitter. That neurotransmitter travels to receptors that are on the postsynaptic neuron, and they they deliver a message there. And then in green, you have a process from an astrocyte that is kind of wrapped around this synapse. So some of these neurotransmitters that are released by the presynaptic neuron are actually binding to receptors that are found on the astrocytes. And the astrocyte itself is able to release its own transmitters that are shown in purple here. We wouldn't call them neurotransmitters because they don't come from a neuron. We would probably call them astrotransmitters. But these transmitters could also have receptors on the presynaptic neuron or on the postsynaptic neuron, and so they can alter the way that this synapse is functioning. And that ends up being really important for neuronal plasticity and for signaling in the brain. So astrocytes are luckily like the most complicated type of glial cell, and the other ones are a bit more simpler. So after astrocytes in the central nervous system, you have oligodendrocytes, which basically their job is to make myelin sheaths. So they are little cells, like you can see one here. They send processes out, and those processes will wrap around axons that are kind of traveling by the oligodendrocyte, and those processes where they're wrapped around the axon are going to form myelin sheaths so that this axon can send messages faster. Here you have another view of an oligodendrocyte. The cell body is in the center, and it has a bunch of processes coming out from it. And you can see at the end of a lot of these processes, they're kind of like widening, getting bigger. That's because they are wrapping around an axon that isn't shown just because it's not stained, and they are laying down myelin there. You also have microglia in the central nervous system, which are kind of the immune cells of the brain. So most immune cells kind of travel in the blood, and you also have kind of immune cells that are like resident in different tissues. But in the brain, you don't have like proper immune cells that belong to the actual immune system. You have microglia instead. Just, immunity for the brain. So microglia are phagocytes, which means they are cells that eat stuff. Phago means eating. So they could eat a bunch of different stuff. If you get microbes coming into the brain, that shouldn't really happen because they should be kept out by the blood-brain barrier. But it might happen. Sometimes there's a whoopsie. So if you end up with microbes in the brain, the microglia will be able to eat them. Also, anytime that neurons die or glial cells die, a microglia will come and eat them up and recycle their components. And actually, unused synapses they will eat as well. So You might have heard the phrase like use it or lose it when it comes to the brain. So if you have connections in there that you are not using, you will actually lose them because the microglia are going to come by and eat them, if they're not being used. Microglia also can cause inflammation in the brain. If sometimes that might be necessary. Of course, sometimes that can also get out of hand and be bad for you in the end. And they also do other stuff as well. So we definitely don't know all of the functions of microglia. Microglia are kind of interesting cells because they can actually change their shape, and they have a couple different forms. So they have kind of like an inactive form and an active form. When they're inactive, they have a lot of processes coming out from the cell body. So that would be shown kind of here, like all these guys on the left are kind of more inactive microglia, and you can see the main cell body and all the processes coming out from it. This view is also showing those inactive microglial cells. So in green, you can see the cell bodies for all those cells, and then you can also see these little green threads, which are the processes coming out from them. So when they're inactive, which would be like under normal conditions, they're going to have all these processes coming out, and the processes will actually be moving around. They'll be moving through the tissue of the brain or the spinal cord and looking for problems, basically. And if they find a problem, then the microglial cell is going to activate and change its shape to be more like a blob. So like here is a microglial cell that is kind of in the process of activating, and here you have one that's fully activated. So it's just going to blob up and it'll go deal with that problem. Then the last type of glial cell that you have in the central nervous system is the ependymal cell. Ependymal cells line ventricles in the brain. Ventricles are just kind of empty spaces in the brain that are filled with fluid. So, epithelium always lines surfaces. In the brain, you don't have like proper epithelium. Instead, you have the ependymal cells lining those surfaces. And they form what we call the neuroepithelium. So it's like an epithelium, but it's in the brain or in the spinal cord. It basically looks a lot like epithelium. The ependymal cells have cilia on their apical surfaces, and their shape is going to be either like simple cuboidal epithelium or it'll be like simple columnar epithelium. But we don't say that it is epithelium because it doesn't come from the same tissue or the same embryonic tissue that epithelial tissue comes from. It actually comes from glial cells. So if you have like a stem cell that's dividing and differentiating to become a glial cell, first it'll go like down the glial cell path and be committed to becoming a glial cell, and then it'll form maybe into an ependymal cell. So when you look at where their cells are coming from, ependymal cells come from glial cells. They don't come from epithelial cells. So in this image here, you can see the ependymal cells forming the neuroepithelium. These ones look like simple columnar cells, and you can see the little brush on the top of it. That's the cilia that those cells have. So these cells would be found lining ventricles in the brain or lining the central canal in the spinal cord. So in this image of the brain, you can see those ventricles in blue, just hollow spaces that are filled up with fluid. And it actually goes down into the spinal cord as well. So in the next chapter, when we talk about the brain and the spinal cord, then we'll talk more about the ventricles. But they're going to be filled up with this fluid that's called cerebrospinal fluid, or CSF. That fluid is actually going to be made by the ependymal cells. And then they're also going to be circulating it through the ventricular system using their cilia. So when their cilia beat, it circulates the CSF. The CSF is really important. It kind of helps the brain be buoyant. So we had mentioned that nervous tissue is really weak, and actually the brain is heavy enough to crush itself. So if you just have the brain sitting in the skull with no CSF around it at all, the top of the brain is going to bear down on the bottom of the brain, and all those cells on the bottom are actually going to get crushed and die. So actually, instead of that happening, you have ventricles inside the brain, which give it some buoyancy, and also the brain itself is kind of separated from the skull by a layer of CSF, also providing more buoyancy. Another function of the CSF is to provide shock absorption. So anytime you get kind of a blow to the head, any shock that is coming through the brain will hopefully be absorbed by the cerebrospinal fluid instead of being absorbed by the nervous tissue itself, which would damage that tissue. And finally, you can have waste that are released into the CSF by neurons as they're functioning, and that CSF is actually going to be removed from the brain, and it'll end up joining back with the blood, and so those waste products will also end up being drained into the blood. So here you have just kind of a summary image of the glial cells that you would find in the central nervous system. So in yellow here you have neurons themselves with their myelinated axons coming off of them. In green you have the astrocytes. You can see they're sending out a lot of processes. Some of those processes are contacting neurons, helping to anchor them in place. Some of those processes are contacting capillaries. So here you have the astrocytic infeet wrapped around those capillaries that help form the blood-brain barrier. You would also have some of these astrocytes participating in synapses. Here you actually have an astrocyte that's contacting a dendrite, so we might expect to have a synapse there that's just not shown, and the astrocyte would be helping to regulate signaling that's occurring at that synapse and plasticity, and also recycle stuff that's released from the synapse. Then you have in blue the oligodendrocytes, and you can see them sending out processes that wrap around axons, and they're laying down myelin sheaths there. In red, you have the microglia that are serving as immune cells. These ones you can tell are inactivated because they have all the little processes coming off, so they're scanning for some type of problem. If they find a problem, they will blob up and go deal with it. So that might mean that they end up eating something, a dead cell or an unused synapse or a microbe. Might mean that they end up causing some inflammation or doing something else. And then finally, here you have a ventricle in the brain, and in kind of pink, you have ependymal cells that are lining it, and you can see their cilia. These particular ependymal cells are actually producing something that would be CSF actually, and then the cilia are going to be circulating it around in the ventricle. Next, we'll cover the glial cells that you find in the peripheral nervous system, starting with the Schwann cells. So Schwann cells are pretty similar to oligodendrocytes in the central nervous system. So they're also going to be laying down myelin sheaths around axons, but their shape is different, so they don't have processes. Instead, it's more like the entire cell is just wrapped around the axon, and the membrane of the Schwann cell is going to be full of fatty substances that actually form the insulation of the myelin sheath. So you're going to find those Schwann cells wrapped around most of the axons in the peripheral nervous system because most of those axons are myelinated. Most axons in the peripheral nervous system are pretty long because they're traveling along, basically the whole length of a nerve in general. And you need to basically send those messages real fast. So for instance, if you're like touching a hot stove, you need to get that pain information real frickin' fast, and then you need to send a motor signal out to jerk your hand away also really fast. Because the longer that your hand is on this burner, the worse you're going to get burned. So whenever you encounter kind of danger like that, you need to be able to react to it really, really quickly so that you won't be damaged. Also, if you're trying to do like sports or anything that's involving like precise coordination, if this baseball player or batter isn't able to hit the if his PNS neurons are not like myelinated, if they're sending messages slowly, then he might see the ball coming and send messages out to his muscles to move the bat, but they're going to get there late, and he's going to swing the bat late, and he's not going to hit the ball. So messages need to travel really quickly in the PNS in order to let you behave in a way that is relevant and kind of helps you actually do the task you're trying to do and keep yourself safe. So that's why most of those axons are myelinated. You do have a few exceptions. There's a few axons in the peripheral nervous system that are short, so they don't need any myelin around them. And there's also some types of axons that send pain that are not myelinated. So whenever you feel pain, if you let's say you stub your toe on something, first you're going to get that really sharp pain, right? So that's traveling in on a myelinated axon, and you feel it basically immediately. But after that, you're going to get kind of a low throbbing pain coming in. That signal is coming in on an unmyelinated axon that is more slow. Then the last type of glial cell that we have in the peripheral nervous system is the satellite cell, which functionally is kind of similar to astrocytes in a way, although it's even less well understood than astrocytes are. So we really know very little about satellite cells and what they do, but it is becoming clear that they do a lot of things. So these are kind of small cells that you would find wrapped around or surrounding cell bodies in the peripheral nervous system. And we think that they're basically kind of regulating the extracellular matrix and making sure that the neurons are protected from any toxic substances or chemicals that are in the extracellular matrix. They also might be providing those neurons with nutrients and with structural support. And we've actually found that they're able to communicate with each other using gap junctions. So some of them will be connected using gap junctions, and they can make sure that they're coordinating their functions that way. So here you have a couple images of neurons surrounded by satellite cells. The satellite cells are the little dark blobs, and the neurons are the much larger things. So these would be neuronal cell bodies in the peripheral nervous system, and then the little dark blobs around them are the satellite cells. So here, again, just a summary of the glial cells in the peripheral nervous system. So you have your Schwann cells that are wrapped around axons, wrapped around most of the axons in the PNS, and they are, basically forming myelin sheaths to make messages travel faster in the peripheral nervous system. And then in your cell bodies, you're going to have satellite cells wrapped around them, which would be doing various things, providing some support maybe, providing nutrients, and protecting them from toxic chemicals. So we'll just talk a little bit more about myelin sheaths before we move on. Every myelin sheath is actually a bit of cell membrane that's wrapped around an axon many, many, many times. And the membrane there is actually going to be extra fatty. Of course, membranes are already made out of, not lipids precisely, but chemicals that are really similar to lipids. They're definitely quite hydrophobic. And then you're adding extra lipids to those membranes. So they're really, really, really fatty. You actually don't have any cytoplasm in there. It's just layers of membrane wrapped back to back to back. And they can be really thick. You're not going to have any ions that are able to cross the myelin sheath because it is so hydrophobic, and ions have a charge, so they're really hydrophilic. They're not going to interact with anything that is hydrophobic. They could only cross a membrane if there's a channel in it, basically, and in a myelin sheath there's going to be no channel and also really, really thick layers of membrane, so it's impossible for ions to get across it. Ions are actually really important for sending electrical signals in neurons. So when you have local potentials traveling in a dendrite, or when you have action potentials traveling in an axon, that means that actually you have ions crossing the membrane. Since an ion has a charge, when it moves from one place to another, it causes an electrical current. An electrical current is just charge moving from one place to another. So in order to prevent electrical current from crossing the membrane, where it's insulated, you would just have a myelin sheath so that the ions can't get across. Whenever you have myelin sheaths, each one is just like a little band, and then you have a gap, then you have the next one. Those little gaps are called the nodes of Ranvier, and those end up really being really important for actually sending messages down the myelinated axon. So normally when you have an action potential traveling down the axon, it'll just be traveling from one bit of the membrane to the next bit of the membrane to the next bit of the membrane to the next bit of the membrane, so on and so forth. But when you have a myelinated axon, it's actually going to hop from one node of Ranvier to the next one. So just hop from node to node to node, and it'll skip the whole myelin sheath. And that makes it go way faster. Actually, if you have no myelin sheath around your axon, so if it's unmyelinated, then the speed at which it sends action potentials might be anywhere from half a meter per second up to 10 meters per second. But if you put some myelin around that axon, now it can send messages at anywhere up to 150 meters per second, which is crazy, crazy fast. So it makes a huge difference. And then this image here is just showing white matter versus gray matter in the brain. This is actually a section of a rat brain. The kind of tan bits are the gray matter, and then the white bits are the white matter. That white matter is actually formed just by myelin sheaths. So wherever you see a band of white matter, that lets you know you have a bunch of axons traveling through there, and they're all myelinated. So it's actually the fat in the myelin that is causing this white color to form. It's just like looking at fat and meat. It's real white. It's the same thing here. Wherever you see, not fat, then you don't have a lot of myelin. That lets you know there's not a lot of myelin in there. We call that gray matter, even though it kind of looks tan here. If you were to slice this real thinly, then it would start to look gray. So now we'll get into the details of how neurons use electrical signals and send them to each other. Or I guess just how electrical signals operate in neurons. So first we have the concept of the resting membrane potential. The resting membrane potential is a difference in the electrical charges on one side of the cell membrane versus the other side when that cell is at rest. And an electrical potential is just another word for a voltage. So this is actually a voltage across the cell membrane. So in a battery, of course, every battery has a voltage associated with it. In a battery, you have a positively charged side and a negatively charged side, and the difference between those two charges is the voltage of the battery. And it's the same thing for the resting membrane potential. The difference in charge between one side of the membrane and the other side is the resting membrane potential, or the voltage across the membrane when it's at rest. So actually every cell in the body has a resting membrane potential, but it's usually not like crazy important for regular cells. It's mostly important for neurons and also for muscle cells. What the resting membrane potential is exactly, like what value it has, is actually going to depend on the type of cell that we're talking about. But for neurons, for almost all neurons, the resting membrane potential is -70 millivolts. So it's really small. Like a battery would have its voltage measured, in volts, and in neurons or in cells is measured in millivolts because it's really small. So that -70 millivolts means that the inside of the cell membrane is 70 millivolts more negative than the outside of the cell membrane. So it's always the inside compared to the outside. So if your membrane potential is a negative value, then that means that the inside is negative compared to the outside. And if it's a positive value, that would mean that the inside is positive compared to the outside. The resting membrane potential in neurons is formed basically by leak channels. A leak channel is a type of ion channel that kind of opens and closes at random. So it's not really controlled by anything, just at any given time. Some of the leak channels will be open, some of them will be closed. In neurons, you have leak channels for potassium, and potassium is actually really highly concentrated inside of the cell. It doesn't have a high concentration outside of the cell though. So when you have a potassium leak channel and it is open, that means that potassium is going to be able to cross the cell membrane, going from where it has a high concentration to where it has a low concentration. Normally, potassium would not be able to cross the cell membrane because it has a charge on it, and that makes it very hydrophilic, and only things that are kind of hydrophobic have an easy time crossing cell membranes. It's really hard for anything that has a charge to cross the membrane unless there's an actual channel for it, like a leak channel. So when you have these leak channels for potassium, and potassium is leaving the cell because it's going from where you have a high concentration of it inside the cell to where you have a low concentration of it outside the cell, that means that you have positive charges leaving the cell. And that means that the inside is going to end up a little more negative and the outside will end up a little more positive. And in fact, the inside ends up being 70 millivolts more negative than the outside due to these potassium ions flowing out through the leak channels. Or I guess I should say like mostly mostly due to potassium ions leaving through leak channels. There's other channels that contribute as well, but it's mostly the potassium ions that contribute. So this diagram here is showing the resting membrane potential in a cell. So in kind of orange here, you have a cell membrane, and you have charges, pluses and minuses, throughout the whole cell and throughout the solution that the cell is in. So when we have a resting membrane potential, we have a difference in charge on one side of the membrane versus the other side of the membrane. But if you were to kind of zoom out to look at the whole cell and the fluid around it, all of it is going to be electrically neutral, which means that you have an equal number of positive charges and negative charges. So that means if you have a negative ion, you need to have a positive ion as well. For every negative ion or anion, you also have a positive ion or a cation. That way, the whole thing is electrically neutral and doesn't have an overall charge on it. But even though the whole thing is electrically neutral, like if you were to count up all these pluses and minuses, they're the same number. If you're looking right by the cell membrane, you can see a separation. So that the outside has pluses lined up around it, or positive charges all around it, and then the inside has negative charges around it. So the resting membrane potential is kind of where all neurons are starting off at. Anytime you're going to have an electrical signal in a neuron, that means that you need to change the membrane potential. You need to take it away from the resting value. It could either go above the resting membrane potential or it could go below the resting membrane potential. Either way, just any change in the resting membrane potential of a neuron is going to be an electrical signal that's traveling in that neuron. Any change that you have to the resting membrane potential or to the membrane potential of a neuron is going to be caused by ions crossing the membrane. So ions, of course, have a charge on them. That means that anytime they're moving, a charge is moving, and a moving charge is a current. So anytime the ions are crossing the membrane, that means that you have a current, an electrical current flowing across the membrane, and that is going to change the membrane potential, and that is the electrical signal. Ions crossing the membrane is going to rely on the concentration gradients for those ions. So some ions have a high concentration inside the cell and some have a high concentration outside of the cell. Actually, the ions that have a high concentration outside the cell would be sodium, chloride, and calcium. And it's just one main ion that has a high concentration inside the cell, which would be potassium. So you also have like negatively charged proteins or positively charged proteins inside the cell, that also are like charges that are contributing to electrical neutrality in the cell, but they're not able to cross the membrane. They're just too big. So they don't really contribute to electrical signaling. So the molecules that actually do contribute that we're worried about would be just these four ions: sodium, chloride, calcium, and potassium. And actually the most important ones there are just the sodium and the potassium. So sodium, chloride, and calcium all have a higher concentration outside the cell, and potassium has a higher concentration inside the cell. Whenever those ions are crossing the membrane, they're going to flow down their concentration gradient. So that means if you have a channel that opens for sodium that has a high concentration outside the cell, it's going to flow down its concentration gradient and go into the cell. Same thing for chloride or for calcium. But if you opened a channel for potassium, that one has a high concentration inside the cell. So when it flows down its concentration gradient, it's going to end up going outside the cell to where it has a lower concentration. So those ions are only moving because of their concentration gradients. So in order to have electrical signaling in neurons, you need ions crossing the membrane and you need them to have concentration gradients that will cause them to cross. So it's really important for the brain that the concentrations of ions on either side of the membrane are maintained. And that's maintained in large part by the sodium-potassium pump. Which you probably learned about in general biology, but the sodium-potassium pump is going to take 3 sodiums from inside the cell and pump them out, and at the same time it'll take 2 potassiums from outside the cell and pump them in. So that means that it's moving those ions against their concentration gradient towards the side of the membrane where you have more of them already, which means that it's going to cost ATP to do that. It doesn't happen for free. So that pump is really, really important for neurons. It's actually important for all cells as well, but for neurons, you know, they will not be able to function if they don't have that pump running, because the ion concentrations will eventually kind of be lost as ions are crossing the membrane, and at that point you wouldn't be able to send electrical signals anymore. So here you have just kind of an illustration of the way that sodium-potassium pump works. It picks up 3 sodiums from inside the cell. It also uses ATP. It drops those sodiums off outside of the cell, and at the same time it picks up a couple of potassiums from outside the cell and then drops them off inside the cell. Then this other diagram is just kind of showing those ion concentration gradients for sodium, potassium, and chloride. It's actually not showing calcium. So sodium and chloride have a really high concentration outside of the cell membrane, and then potassium has a really high concentration inside of the cell. So if you're looking at outside of the cell, your main charges that you have there are going to be sodium and chloride, so they're basically balanced with each other. You also have some proteins in the extracellular matrix that have charge that are also balanced with those ions. If you're looking inside the cell, the main ion that you have is just potassium, which is all positively charged. So then you need negative charges to balance that out so the cell can be electrically neutral, and those are going to be basically, for the most part, proteins. So you have large proteins that have negative charges on them that are balancing out the positive charges from the potassium. And here it's shown with the sodium ions kind of lining up right by the membrane, and some of these negatively charged proteins lining up right by the membrane to indicate the resting membrane potential, where you have the inside being more negatively charged compared to the outside. And that charge difference is only just right there at the membrane. If you go further away from the membrane, you're not going to see a charge difference anymore. Right, so the only ion that's not shown on here would be calcium, which is more concentrated outside the cell, but it doesn't have like a high concentration. There's only a little bit of it outside the cell. It's just that there's like almost none inside the cell, except for calcium that's kind of stashed in storage. So I mentioned that ions, if ions are going to cross the membrane, they have to have an ion channel that will take them across, or kind of allow them to cross, provide a channel for them to cross. So in neurons, we have 4 types of ion channels that are important for the functioning of the neuron. First, you have ligand-gated channels. That means that you have a chemical that controls whether the channel is open or closed, and it would be like one specific chemical, like, for instance, a neurotransmitter, perhaps. So anytime you have that chemical, which is called the ligand, present, it will bind to the channel and the channel will open. Then if it's not present, the channel will be closed. Ligand-gated channels would be primarily found in dendrites. They can be found in other parts of a neuron, like the cell body, or even occasionally in axons, but they're mostly found in the dendrites. Then you also have voltage-gated channels, which are gated by voltage. So whenever the membrane potential reaches a particular value, the channel will open, and whenever it kind of goes below that value, the channel will close. So of course the value is going to depend on which ion channel specifically we're talking about. So for instance, some of those channels are gated at negative 55 millivolts, which means that anytime the membrane potential gets to negative 55 millivolts or goes above that value, the channel will open. And anytime it drops below negative 55 millivolts, the channel will be closed, which means that it would be closed at rest because the resting potential is negative 70 millivolts. So voltage-gated channels are only found in axons. You can't find them in dendrites or in the cell body of a neuron, only in the axons. Then you have mechanically gated channels. Those are not as important for the brain. You mostly find those in the peripheral nervous system, actually, mostly in sensory neurons. A mechanically gated channel is something that is going to be physically pulled open. So whenever you have like some type of pressure exerted on the membrane, that means that the membrane is going to stretch and the channel will actually be pulled open. Then if you release the pressure, the channel will close back up again. So for instance, like a touch receptor in your skin or a stretch receptor in your stomach, those things are using mechanically gated channels. So whenever you have pressure exerted on the skin, you're going to have pressure on the membranes of the sensory neurons there, and the mechanically gated channels that they have will be physically pulled open. Then finally, you have leak channels. Leak channels just open and close at random, and they can be found in all parts of a neuron. So the dendrites, the cell body, and the axon as well. Their main job is just to help maintain the resting membrane potential. So out of those 4 types of channels, the most important ones for us for understanding electrical signaling in neurons will be the ligand-gated channels and the voltage-gated channels. So those are kind of shown in this figure. On the top you have a ligand-gated channel. On the bottom you have a voltage-gated channel. So for the top one, this is going to be a channel that allows sodium and potassium ions to cross it. And it has a little pocket on it for its ligand to bind. And whenever the ligand binds to that little pocket, the channel will open and sodium and potassium will be able to cross. Each of them is going to cross going down their concentration gradient, which means sodium is going to go from where you have a lot outside to where you have a little, which is inside. And potassium will be doing the same thing, but in the opposite direction. So for potassium, you have a lot inside and a little bit outside, which means potassium is going to flow out. On the bottom, you have a voltage-gated channel for sodium. So it's going to be closed at most, it'll be closed at rest, at the resting membrane potential, and then for this particular channel, it's going to be gated at negative 55 millivolts. So whenever the membrane potential rises to negative 55 millivolts or above, the channel will open and sodium will be able to cross. So anytime that you have ions crossing the membrane down their concentration gradient, moving through an ion channel, you're going to have the membrane potential changing somehow. So it's going to leave the resting membrane potential value and go to a different value. And we have a few, 3 different terms that describe ways that the membrane potential can change. First, you have depolarization. Depolarization just means that the membrane potential is going more positive. So it starts at negative 70 millivolts at rest. If it goes above that value at all, that's depolarization. That can happen anytime that cations enter the cell. So a cation is just a positively charged ion. So if you have sodium or calcium entering the cell, that's going to cause depolarization. But almost always it's going to be sodium. Sodium is like the #1 ion that's associated with depolarization. And anytime that sodium is crossing the membrane, it's always going to be going from out to in because that's the way its concentration gradient points, which means it's always going to cause depolarization. Once you have depolarized the membrane, taking it to a positive potential, or at least a more positive potential compared to the resting potential, then you can have repolarization occur. So in repolarization, the membrane potential starts more positive than the resting value, and it goes back down to that resting membrane potential. So that means that you can only have repolarization if you had depolarization first. So the resting membrane potential is negative 70 millivolts. If you go up from that to negative 30 millivolts, then that is depolarization. And then if you're at negative 30 millivolts and you go back down to negative 70 millivolts, that would be repolarization. Repolarization can happen in a couple of different ways. It'll happen anytime that anions enter the cell, and anion is just a negatively charged ion. So if negative charges are entering the cell, then the membrane potential would go to a more negative value. And also it'll happen if you have cations leaving the cell. So if you have positively charged ions leaving the cell, that means that you're losing positive charges from the inside of the cell and adding them to the outside, which means that the inside is becoming more negative. So the membrane potential would be dropping, in this case dropping back down to the resting value. The main anion that would be able to enter the cell was chloride, and the main cation, well, I should say the only cation that would be able to leave the cell is potassium, which means that sodium is associated with depolarization and chloride or potassium are associated with repolarization, but they're also associated with hyperpolarization, which is the last way that the membrane potential can change. So in hyperpolarization, you're starting at the resting membrane potential and you're just going down from there, just having the potential become more negative. So the same ions that cause repolarization will also cause hyperpolarization. The only difference is that in repolarization, you start from a depolarized value that's above the resting membrane potential, and then you go back down to the resting membrane potential, whereas in hyperpolarization, you're starting at the resting value, and then you're going down from there. So here is kind of a figure showing depolarization. So we start at rest. Here's our cell membrane. This is the inside of the cell, and in blue we have the outside. This blue protein is our ion channel. It is closed currently, and we have little charges indicating the polarization of the membrane, or I guess the membrane potential. So it's starting at rest, negative 70 millivolts, which means the inside is more negative and the outside is more positive. And then here we have an electrode that's inserted into the cell and another one that's placed outside so that we can actually measure that membrane potential. And it's measured at negative 70 millivolts, so we know it's at rest. This is actually a ligand-gated ion channel. So if we have the ligand bind to it, then the channel is going to open. And now sodium ions are able to go through it because this is a sodium channel. So sodium has a high concentration outside the cell. When the channel for it opens, it's always going to flow into the cell down its concentration gradient. That means you have positive charges entering the cell, and that means that the inside of the cell will be less negative and more positive. So now the membrane potential right by this channel is going to start changing and going to a more positive value. And here it's actually going up to negative 60 millivolts. So if we were to graph that, we're starting at the resting membrane potential, and then when the channel opens, we go up to negative 60 millivolts. So that is depolarization. And then here we have a figure showing hyperpolarization and the couple of different ways that it can occur. So again, over here we're starting at rest. We have our ion channel. This might be an ion channel for chloride, or it might be for potassium. It's again a ligand-gated ion channel, and the cell membrane is at rest or negative 70 millivolts. Then if that channel opens, let's say that it is a potassium channel and it opens, because the ligand comes and binds it, now you're going to have potassium leaving the cell. Potassium has a higher concentration inside, so it's going to go down its concentration gradient and flow outside of the cell, which means that you are subtracting positive charges from the inside of the cell and adding them to the outside. That means that the inside is going to end up even more negative compared to the outside than it already was. So you should see the membrane potential go down. In this case, it is going down to negative 80 millivolts. On the other hand, if that ion channel had been for chloride instead of potassium, then when it opens, you're going to have chloride entering the cell because chloride has a higher concentration outside the cell, so it's going to flow down its concentration gradient and enter the cell. That means you have negative charges that are being added to the inside of the cell, so the inside will get more negative. And again, that means that the membrane potential is going to drop, in this case to negative 80 millivolts. And if you were to graph that, you're starting at negative 70, then when those channels open, the membrane potential drops to negative 80, and that is hyperpolarization. So we had already kind of mentioned that there's two basic types of electrical signals in neurons, which are the local potentials and the action potentials. So we'll just kind of compare their characteristics, and then we'll go through each type in more detail. So first, the local potentials, they can also be called graded potentials. They're found in the dendrites of neurons and also in the cell body. So they usually are going to begin in the dendrites and then travel down the dendrites to the cell body and then across the cell body. Local potentials are always going to be caused by neurotransmitters that are binding to the receptors in the dendrites, or occasionally in the cell body. So usually you're going to have ligand-gated ion channels that are serving as receptors for neurotransmitters, and the neurotransmitter would actually be the ligand for that channel. So you have this neurotransmitter receptor in the dendrite of the neuron, and it is also a ligand-gated ion channel. So when the neurotransmitter comes and binds to it, the channel is going to open, and now you're going to have ions flowing through the channel, and that causes your local potential to occur. That's the main way that they occur. You can also have, in certain neurons, mechanically gated channels. That would be just like for sensory neurons, though. Local potentials are called local potentials because they don't make it very far. As they're traveling along the dendrites and across the cell body, they're actually getting smaller and smaller and smaller. So they usually start off like, kind of small, and then they just get smaller as they go. So they're not going to make it all that far, really. If they have a really far distance to travel, they'll probably be gone by the time they get to the end of it. So that's why we call them local. They can either be depolarizing or hyperpolarizing, so they can either take the membrane potential above the resting value or take it below the resting value. And they can have kind of different strengths. So they're usually kind of on the smaller side, but they could be like really small or, kind of medium small or even kind of a bigger one sometimes you can have. So they can be all different kind of intensities or current strengths. And that's why we call them graded potentials, because they're not all the same strength. They can be a lot of different strengths. So your local potentials are going to be, starting in the dendrites, traveling down the dendrites to the cell body. As they travel, they get weaker and weaker and weaker. As they travel across the cell body, they're going to continue getting weaker, and eventually they get to the axon hillock, which is the base of the axon. So you're going to have local potentials coming in all over the cell, going to the axon hillock. So you'll have, all the dendrites will have like local potentials traveling on them, going to the cell body, going across the cell body, ending up at the axon hillock. And at the axon hillock, they're all going to add up together. And when they add up together, they can get to be big enough to actually trigger an action potential in that axon. So the action potential is only going to be found in axons. It's always going to be caused by local potentials. And it involves voltage-gated channels only. So when these local potentials come to the axon hillock and add together, they're going to be changing the membrane potential, and if they change it enough, they can actually end up opening a voltage-gated ion channel in that axon, and that will kind of begin the action potential. Once the action potential has begun, it's going to travel along the whole axon and all the branches of the axon without losing any strength at all, which means it's actually being actively maintained as it travels. If it wasn't being actively maintained, it would end up decaying the same way that a local potential does. So you're always kind of regenerating the action potential as it moves into a new section of the membrane, which we'll kind of go over, later when we talk in more detail about action potentials. Every action potential always looks the same. It has the same three phases every time, depolarization, and then repolarization, and then hyperpolarization. So first you depolarize the membrane, taking it more positive than rest, then you repolarize it, taking it back to the resting value, and then you hyperpolarize it, taking it below the resting value. Action potentials are always the same strength as well, which is, strong. So when we are depolarizing the membrane in an action potential, we always take it up to the exact same value every single time. And then finally, when action potentials get to the end of the axon, they will cause neurotransmitters to be released from the axon terminals. And then those neurotransmitters are going to go across the synaptic cleft and bind their receptors on the next neuron and start some local potentials of their own, and the kind of cycle will continue. So now we'll go over the details of local potentials. So first you have just the fact that local potentials don't travel that far because as they travel, they decay, or they kind of get weaker in terms of the strength of the current. So here you have just kind of a couple of figures that are illustrating that. On this first one here, we see an axon coming in to contact a neuronal cell body in this case. And so you have a synapse right here. When action potentials come down this axon, they're going to cause neurotransmitters to be released, and those transmitters will cross the synaptic cleft and bind to the receptor, which in this case is a ligand-gated ion channel for sodium. So when they bind to that channel, they're going to open it, and then sodium will flow through. So sodium is going to be entering the cell. You have positive charges entering the cell, so that means you're going to have depolarization. You're going to have a depolarizing local potential right here where that channel is. If you're going to graph that local potential, it might look something like this. This is just, I guess, the local potential and also its spread. So on the x-axis here, you have the distance from the stimulus, which is in the middle, and on the y-axis, you have the strength of the current, or the strength of the local potential, or I guess the amount of change in the membrane potential that is caused by sodium entering the cell. So, right here in the middle, you have the actual point of origin, which would be this ion channel, and then if you go a little further away to the 1, that corresponds to these ones right here, so it's a little bit further from that ion channel. The 2 is even a little bit more further, and the 3 even more further, and the 4 and the 5 are even farther away. So each number is getting farther and farther away from the ion channel that started this local potential. And you can see that the strength of the local potential is getting smaller and smaller as you go further and further away from the ion channel where it started. So by the time you get to 0.5, which is all the way out here, you're already basically back to the resting membrane potential value. The reason that is because you just have these ions flowing through this particular ion channel, and then they're going to start spreading out once they get into the cell. So right by that ion channel where they're first rushing in, you have kind of a high concentration of them. So there's a lot of positive charges right there. That's going to mean that you have a stronger depolarization right there. But as you're getting further away and those ions are spreading out, they're less concentrated, which means you have less positive charges or more spread out positive charges, and the effect on the membrane potential overall is going to be less and less as they spread out more and more. The same basic thing is shown in these graphs here. Here's your ion channel that is starting the local potential. This is again a sodium channel, so when it opens, sodium is going to come into the cell, and then those ions are going to start spreading out from there. So they're going to be diffusing along the cell, on the interior of the cell. And then here you can graph the effect on the membrane potential that that channel has when it opens. So when it first opens, right by the channel, you have a significant depolarization, and as you go further away from the channel on either side, the depolarization gets smaller and smaller. This is for kind of a strong stimulus in the first place. If you had a smaller stimulus so that the channel was open for maybe a shorter period of time, then you would start with a smaller depolarization and it would still decay as you get further and further away from it. So this is yet one more slide showing how local potentials decay as they travel. So in this case, in this figure, we have an axon terminal contacting a neuron at its cell body in this case, and you are going to be releasing neurotransmitters from this axon terminal. They cross the synaptic cleft and bind to a receptor. That receptor is probably a sodium channel, so it's going to open that channel and then sodium will enter the cell, and that causes a local potential. And as the sodium ions travel diffusing through the cell, they get more and more spread out, more and more diffuse, and the effect they have on the membrane potential is going to go down steadily. So if you measure the effect on the membrane potential that those sodium ions are having right next to the ion channel, you can see that they're having a pretty significant effect, taking it from rest all the way up to negative 40 millivolts. Negative 55 millivolts is the threshold for triggering an action potential, but only if it's actually at the axon hillock. Then if you measure further away from that ion channel, you're going to see that the effect on the membrane potential is smaller. So now we're barely getting up to negative 55 millivolts. And by the time you actually get to the axon hillock, that potential has decayed even further. So now it's not even big enough to actually reach the axon hillock. It's not even big enough to actually reach the threshold value of negative 55 millivolts. If it does not get up to negative 55 millivolts, that means you are not going to have an action potential firing. It's just not strong enough. Here you have basically the same situation, except now you're starting with an even stronger local potential. That might happen because you really had a lot of neurotransmitter released from the synapse, which means that this ion channel here is going to spend a lot of time open, and actually in reality there's probably more than one ion channel there, so you're going to be opening multiple of them at the same time if you have extra neurotransmitter. And so now we see that our local potential, when we measure it right by the ion channels, is getting even higher, even more depolarized than -40 millivolts. Maybe it's -30 or something. And it still decays as you measure further and further away from that ion channel, but when you measure at the axon hillock, it actually is still barely enough to get over the threshold value of -55 millivolts. So now it actually is going to be strong enough to trigger an action potential to travel down this axon. So usually when you have local potentials reaching the axon hillock and being enough to trigger an action potential, it's usually not just one that ended up being really strong, so that it was still strong enough by the time it got to the axon hillock to actually start that action potential. Usually you're going to have multiple local potentials kind of coming in. So they're always going to be coming into the axon hillock, and sometimes they're going to be adding up to be enough to actually take the membrane potential at the axon hillock from -70 millivolts up to -55. And when you get to -55 millivolts, that's when you trigger the action potential. So all the local potentials coming into the axon hillock are going to kind of figure into that equation. They're all going to add up. And then you just kind of see like where they take the membrane potential to, and if it's enough to make an action potential or if it is not. Different local potentials, though, will kind of have a different impact on you know that addition. So if you have a local potential that's starting kind of close to the base of a dendrite or on the cell body itself, that's probably going to be stronger by the time it gets to the axon hillock, and it's going to have a larger effect on the ability of the neuron to fire an action potential. If you have a local potential that started way at the end of a dendrite, then by the time it gets to the cell body, it's going to be really weak, maybe even gone already. So it's not going to be having much effect on the ability of that neuron to fire an action potential. It also depends on how strong the stimulus is that begins the local potential. So if you're releasing a lot of neurotransmitter, you're going to end up with a bigger local potential that will travel farther and have a bigger impact on the membrane potential at the axon hillock. If you have just a little bit of neurotransmitter released, then you're going to get a smaller local potential that doesn't travel as far. Whenever you have more neurotransmitter present, that means you're going to have more of those ligand-gated ion channels open. When you have more of those ligand-gated ion channels open, that means you have more ions coming into the cell or leaving the cell, depending on what ion it is. But you know, we're usually talking about sodium, so you would have more sodium coming into the cell if you have more ligand-gated ion channels open for it, and that would mean you need more neurotransmitter to be released. Another thing that affects it is how long the stimulus lasts that is actually causing this local potential. So basically, are you releasing like one small burst of neurotransmitter, or are you kind of releasing neurotransmitter over an extended period of time? The more neurotransmitter you release, and for the longer period of time, the stronger the local potential that you're going to get. If you get a lot of neurotransmitter released for kind of a long period of time, then you're going to get a local potential that ends up being pretty big and also kind of last longer in time, and then it might be able to overlap with more other local potentials. So if you have just kind of a small brief local potential, then it's really hard for it to end up coming to the axon hillock at the exact same time as another one, so they can actually add up together. But if you have kind of a longer lasting local potential, then it'll be at the axon hillock for longer, and there's just a longer or a larger window of time for another local potential to show up and add up with it. Then finally, it depends on which channels are opening exactly. So, you know, what ions are these channels allowing to cross? And then for those ions, like how much of a concentration gradient do you have for them? Is it just a small gradient or is it a big gradient? And which way is that gradient pointing? So If we're talking about sodium, that's positively charged, so it's going to be entering the cell, bringing right, well, I should say is positively charged, and it has, a higher concentration outside the cell, which means it's going to enter the cell. So it's bringing positive charges into the cell, and it's going to depolarize. There's also quite a strong concentration gradient for sodium, which means you have like a whole lot of it outside the cell and only a tiny little bit inside the cell, a really big difference in terms of how much you have on either side. So that's a steep concentration gradient. Whenever you have sodium flowing down that gradient, it's going to move really fast. You're going to get a lot of sodium crossing the membrane even in a short period of time. If you compare that to calcium, calcium has like very little inside the cell, but it also doesn't have like that much outside the cell. It's kind of a small concentration on both sides of the membrane, just a little bit bigger outside than inside. So when you open channels for calcium, you're going to have calcium coming in, but it's not going to go in really fast. It's going to kind of dawdle in, moving really slow, which means that you're going to get less of it coming into the cell per unit of time that its channels are open compared to sodium. That means you're going to get a smaller local potential that's going to have a smaller effect on the axon hillock. And then again, you have to consider like what type of ion this is. So if it's sodium or calcium, that's going to be a positive ion entering the cell, depolarizing it, taking it closer to threshold at the axon hillock. On the other hand, if you had potassium leaving the cell, or if you had chloride entering the cell, chloride is negatively charged, so when it enters, the cell goes more negative, and potassium is positively charged, but it always leaves because that's the way its concentration gradient points. So when it leaves, you're losing positive charges from the inside, and you're taking the cell again more negative. So if you have local potentials that involve either one of those ions, those are actually going to be hyperpolarizing potentials, which means that you're taking the cell further away from firing an action potential. When those chloride or potassium local potentials actually reach the axon hillock, they don't take it closer to the threshold of -55, they take it farther away from threshold. They make it less likely for an action potential to occur. So all these factors kind of affect like if the local potentials will actually add up to be strong enough to take the resting membrane potential from -70 to -55 millivolts and actually fire an action potential. So now we'll just kind of look at how those local potentials are adding up. So here you have a diagram of a neuron being contacted by another neuron. So when you have activity in this first neuron and action potentials traveling along its axon, when they get to the end, they will cause neurotransmitter to be released that will cross a synaptic cleft and bind to the receptors on this dendrite, and that will cause local potentials to occur in this neuron. Those local potentials will travel down the dendrite and across the cell body to the axon hillock, where they might begin an action potential. So you can see kind of the electrical graph here of those potentials. So on this kind of side of the graph, on the left side, you have your local potentials that are being recorded in these dendrites. You can see some of them are smaller, some of them are bigger, some of them are depolarizing, some of them are hyperpolarizing. And then when they add up at the axon hillock to take the membrane potential to -55 millivolts or above, then an action potential will be triggered, and the action potentials will always look the same way. On this diagram, it's showing 3 different dendrites on a neuron, and each of these dendrites is being contacted by another neuron. So you have 3 different synapses here, and we have activity on all of these synapses. So we see the neurotransmitter being released. That neurotransmitter is going to cause local potentials to arise in these dendrites, and all these little kind of black dots here, I guess these are our ligand-gated ion channels that would actually be receptors for these neurotransmitters. So these neurotransmitters are going to bind to these ligand-gated ion channels and open them. Then you're going to have ions crossing the membrane. And it has these little graphs here to show how the membrane potential is changing as these synapses are stimulated. So you can see that you're having depolarization, which means, of course, you've got sodium entering through these ligand-gated ion channels. So when those channels open, sodium comes into the cell, that depolarizes the cell. Sodium is going to start diffusing out into the cell, That means that your local potential will be traveling with that diffusing sodium and getting weaker and weaker as it goes. But you have 3 different synapses here, and they're all active at the same time. So even though your local potentials are getting weaker and weaker as they travel towards the axon hillock, they're also kind of converging with each other, and they're going to end up building on each other. And in this case, they actually do build up to negative 55 millivolts, and that is enough to open these voltage-gated ion channels that are shown here with upside-down triangles. And these voltage-gated channels are going to cause the action potential to occur. They open at negative 55 millivolts. So if your local potentials can all come to the axon hillock at the same time as each other, they may be strong enough, all of them together, to raise the membrane potential from negative 70 millivolts at rest to negative 55 millivolts, and then you have the action potential fire. So in that diagram, we saw multiple synapses being active at the same time. So multiple dendrites had local potentials. That's kind of what's shown on this left-hand graph. You have one neuron receiving messages from 2 other neurons. So you're going to have local potentials coming in at multiple dendrites at the same time, potentially, and adding up. To potentially take the axon hillock to the threshold voltage of negative 55 millivolts. If you graph that out, here we can see the membrane potential is graphed, and we have our activity. We have local potentials happening here. So when this first neuron sends an action potential down its axon and you have activity at that synapse, you're going to get a local potential in your neuron labeled number 3, and that local potential is shown here. So when you have action potentials in neuron 1, that causes a local potential of about this size, and it's about this size when it gets to the axon hillock. So it's taking the membrane potential from negative 70 up to looks like maybe negative 63 or 64 or something. That's not enough to get to threshold. Similar thing for neuron 2, when you have an action potential fire down its axon, you're going to have activity at the synapse, and you're going to have local potentials in this postsynaptic neuron. And those are graphed right here, or that's graphed right here. So when neuron number 2 signals to neuron number 3, then you get a similarly small sized local potential in neuron 3. But if you have both of those synapses active at the same time, so both neuron 1 and neuron 2 are sending action potentials down their axons, then those local potentials actually overlap with each other and add up to be enough to take the membrane potential at the axon hillock up to negative 55 millivolts, and then you fire the action potential. So that's called spatial summation. It's called spatial because you've got different synapses active at the same time, and they're at different places. So spatially, they're apart from each other, and local potentials are coming in from different areas and adding up together when they get to the axon hillock. On the right-hand side, we have temporal summation. In this case, we only have one presynaptic neuron, and we have our postsynaptic neuron. And we have graphs of our local potentials that are coming in from the presynaptic neuron. Well, they are occurring in the postsynaptic neuron in response to stimulation by the presynaptic neuron. And if you start getting faster and faster action potentials on this presynaptic neuron's axon, you're going to get more and more local potentials in the postsynaptic neuron, and they actually might not have time to fully fade away before the next one arrives. So then they're going to start overlapping with each other again, and the membrane potential may rise up high enough to reach threshold, and then you can fire an action potential. So spatial summation is where you have multiple different synapses, local potentials coming in from those different synapses. So they're coming in from different parts of the cell, coming into the axon hillock and adding up together. In temporal summation, you have just one synapse that's active repeatedly. So you have multiple local potentials coming from that synapse, and they start coming so fast that the first one doesn't have a chance to fade before the second one is already there, and they add up together that way at the axon hillock to trigger an action potential. Of course, in real life, we don't just have depolarizing local potentials. We also have hyperpolarizing local potentials. So this diagram here is showing maybe a slightly more realistic neuron that has, plenty of dendrites and lots of other neurons coming in to form synapses with it. So these colored axons would be coming from other neurons. So these are all presynaptic neurons in these colors, and in tan you have your postsynaptic neuron. And you can see that 3 of these synapses are excitatory. So when you have activity at those synapses, it's going to cause a depolarizing local potential, and the membrane potential will rise. And we call that excitatory because you're going closer to having an action potential. But we have one of those synapses that's actually inhibitory, which means that it's going to cause hyperpolarizing local potentials when it's active, and we call that inhibitory because it's taking the cell farther away from firing an action potential. So all these local potentials will be coming in at different times from all these synapses, and also from other ones that aren't being shown in this diagram, and they're all going to converge at the axon hillock. Some of them will be strong, some of them will be small, some of them will be depolarizing and excitatory, some of them will be hyperpolarizing and inhibitory, and they'll all kind of add and minus together to determine if you're going to fire an action potential. And that's also kind of shown in this graph here of the membrane potential at the axon hillock. So you can see it starts at -70, then you start getting some local potentials coming in. These are depolarizing, so they're taking the membrane potential higher, closer to threshold, but then you get an inhibitory potential, and the membrane potential starts to drop. Then for a little while you don't have any potentials at all. You have no local potentials arriving, and then the membrane potential is going to start slowly dipping back down. If you don't have anything else happen, it'll eventually get back to rest. But then you have another depolarizing potential arrive, and then another hyperpolarizing potential, and then finally a larger depolarizing potential that actually takes you to threshold. So whether a neuron actually fires an action potential depends on this really kind of messy addition of all these different local potentials that are arriving at different times. Some of them are depolarizing, some of them are hyperpolarizing, so you actually subtract them instead of adding them to the membrane potential. Yeah, so it's kind of a messy process. So now we'll focus more on action potentials themselves and kind of what exactly is happening in an action potential, how they move down the axon, and stuff like that. So first, action potentials are only using voltage-gated ion channels, as I had mentioned. So you're not going to find those types of ion channels on the dendrites of the cell or the cell body. You find them in axons. The first place that you're going to find those types of channels is in the axon hillock itself. You have to have those voltage-gated ion channels in order to have an action potential. So you have to have them at the axon hillock so you can begin your action potential, and then you need to have them also along the entire length of the axon in order to propagate the action potential. So action potentials are actively maintained by opening new voltage-gated ion channels. So if you don't have those channels in a section of your axon, you're not going to be able to maintain an action potential through that part of the axon. So having those voltage-gated ion channels throughout the whole axon is really important for the action potential's ability to travel along the entire axon without losing its strength. And we've got 2 basic types of voltage-gated ion channel that are important in the action potential: the voltage-gated potassium channel and the voltage-gated sodium channel. This diagram is showing the voltage-gated potassium channel. So it's closed at certain voltages, and it's open at certain voltages, and when it's open, potassium is going to be able to exit the cell down its concentration gradient. That is always going to have a hyperpolarizing or repolarizing effect on the membrane potential. It's always going to make the membrane potential go down because you have positive charges leaving the cell, which means the inside has less positive charges, so it's more negative. The voltage-gated potassium channel is actually gated at -55 millivolts, which means that when the membrane potential rises from rest, -70, up to -55, then the channel is going to be triggered to open. And then when the membrane potential drops back down to -55 or below, then the channel will be triggered to close. But the voltage-gated potassium channel is real slow. So when it opens, it doesn't just like open just like that. It's a whole process. It takes its time. So it's going to take some milliseconds actually for the voltage-gated potassium channel to open, which doesn't sound like a lot of time, but in terms of your cells functioning, milliseconds is actually a lot of time. So it's triggered to open at negative 55 millivolts, but by the time it actually does open, the membrane potential might not be negative 55 anymore. It might have changed from that value. Then we have the voltage-gated sodium channel, which is also a little bit complicated because it actually has 3 states. So you would think about a normal gate as being like either open or closed, but the voltage-gated sodium channel could be 3 things. It could be open, it could be closed, or it could be what I like to call locked. So the book is going to use kind of different names for these states. It's going to call closed resting, the resting state. It's going to call open, the activated state, and it's going to call locked, the inactivated state. But for me, it's just easier to say closed, open, and locked. On the test, I'll actually use both sets of terms, resting, activated, inactivated, and closed, open, locked. So you can just remember whichever set is easier for you. So the way that the voltage-gated sodium channel is able to have these 3 different states is by actually having not just one gate on it, but 2 gates. It has its first kind of regular gate, which is called the activation gate, that is going to open or close at negative 55 millivolts. And then it also has its inactivation gate, which I call the lock gate. So if that gate is closed, then the channel is actually locked. If the regular gate is open, well, I guess I should say, if both gates are open, then the channel is open. If the activation gate is closed, then the channel is closed. If the inactivation gate is closed, then the channel is locked. Or if the lock gate is closed, then the channel is locked. The lock gate or the inactivation gate is also actually voltage sensitive, but it's just gated at a different voltage than the regular gate. The regular gate is gated at negative 55 millivolts, and the inactivation gate is probably gated at around 30 millivolts. So you've got your 3 states for this channel to be in, closed, open, or locked. When it's closed, that means that you're probably starting at the resting membrane potential. Your regular gate, your activation gate, is closed, and the inactivation gate or the lock gate is actually open. And we actually have this diagram here that kind of shows these different states. So on the top we have the resting state. These little tabs that look like they belong to a pinball machine. These are the kind of 2 halves of the regular gate. And then this little ball hanging off of the channel is the lock gate or the inactivation gate. So when you're at rest, this lock gate is open and the regular gate or the activation gate is closed. And the channel itself is closed. Then you can transition to the active state or the open state of this channel, in which the inactivation or the lock gate is still open and the regular gate or the activation gate is also open. So now sodium ions can cross through the channel, and sodium will actually cross always into the cell, going down its concentration gradient, which means it's always going to take the membrane potential more positive because you have positive charges entering the cell. After you've activated this channel and it's in the activation state or the open state, next it's going to transition to the inactivated state or the locked state. In that state, your regular gate, your activation gate, is actually still open, but the inactivation gate or the lock gate has closed. So we call the channel inactivated or locked. Then after that is going to reset and go back to the resting state or the regular closed state. So the voltage-gated sodium channel is always going to cycle through those 3 states. First closed, then open, then locked. Then we'll kind of go through our 3 phases of the action potential, starting with phase 1, which is depolarization. So in the depolarization phase, when you're kind of starting an action potential, you're always going to be at the axon hillock, and you're going to have your local potentials coming into the axon hillock and adding up there. You can also call the axon hillock the trigger zone, and so you have your membrane potential there. It's going to start at negative 70 millivolts. And if your local potentials add up to be big enough, then they're going to take the membrane potential up to negative 55 millivolts, or maybe even higher. Once the membrane potential at the axon hillock reaches negative 55 millivolts, your voltage-gated sodium channels will open. So their regular gate is going to be triggered to open at negative 55 millivolts. So that channel is going to open up. When that happens, you have sodium entering the cell, going down its concentration gradient. And that's going to be depolarizing the membrane potential, and that's the reason why we call this the depolarization phase. As you have sodium coming into the cell, actually what's going to happen is that you're taking the membrane potential even more positive, and even more voltage-gated sodium channels will open. So it's actually a small positive feedback loop. You can start out with opening just a couple of those voltage-gated sodium channels, then as sodium enters a cell and it becomes more and more depolarized, you're going to open more and more of those voltage-gated sodium channels and get even more sodium entering the cell. Actually, you're going to get so much depolarization, so much sodium entering the cell, that the membrane potential is going to go all the way up to 30 millivolts. So at the beginning of the depolarization phase, you would be at negative 55 millivolts, which is where the phase is triggered to begin because your voltage-gated sodium channels will open. At the end of the depolarization phase, you've had so much sodium enter the cell that now your membrane potential is 30 millivolts. And this diagram is just kind of showing that your sodium channels are open, so you can see the lock gate is open and the regular gate is also open. Sodium is flowing into the cell, depolarizing it, and your voltage-gated potassium channels are still closed. So actually, as I had mentioned, these channels are triggered to open at negative 55 millivolts, which means at the very beginning of the depolarization phase, when your sodium channels open, your potassium channels are also triggered to open, but they're very slow. So they don't actually open during the depolarization phase at all. They're just working on it. After you finish the depolarization phase, you move to the next phase, which is repolarization. At the beginning of the repolarization phase, your voltage-gated sodium channels will lock and the voltage-gated potassium channels will open. So that means that on your voltage-gated sodium channels, the regular gate or the activation gate is still open, and your inactivation gate or the lock gate is now closed. That's also a voltage gate, and it's probably gated at 30 millivolts. So probably what happens is that at the very end of the depolarization phase, the membrane potential gets up to 30 millivolts, and then that causes your lock gate on those sodium channels to close. So now those channels are effectively closed. We say that they're locked or inactivated, but they don't let sodium go through them anymore. So now you're not depolarizing the membrane anymore. And instead you have your voltage-gated potassium channels open. They were triggered to open back at negative 55 millivolts, but they took their time, and now they're finally open. So now you have potassium that's able to cross the membrane. It's going to leave the cell, taking its positive charges with it, which means that the inside will be getting less positive and more negative. And of course, it's leaving the cell because you have a lot of it inside and a little bit outside, so it's going to flow down its concentration gradient to go out of the cell. So as this potassium is leaving the cell and the membrane potential is becoming more negative, we actually call that repolarization, because you're starting out from a more positive value and you're going back down. So you're actually going to go back you're starting at negative 30 millivolts at the very beginning of the repolarization phase, and you're going to go all the way back down to negative 70 millivolts, or the resting membrane potential at the end of this phase. Once you get to negative 55 millivolts, so that's kind of when you're almost through the repolarization phase, but you're not totally done with it yet. Once you get back down to negative 55 millivolts, your voltage-gated sodium channels are going to reset, which means they're going to go back to being regular closed. So at negative 55 millivolts, their lock gate is actually going to open, and their regular gate or their activation gate is going to close. So that basically means that they're ready to go again for the next action potential whenever it arrives. And also the other thing that happens at negative 55 millivolts is your voltage-gated potassium channels are again triggered to close. But again, they're very slow, so they're not going to actually close at that point. they're going to continue to be open, which means that the membrane potential continues to drop and goes from -55 back down to -70, which is the resting value. And here you have kind of a diagram of the ion channels during this phase. First you have the sodium channel, so it is locked for most of this phase, which means the regular gate or the activation gate is still open, but this lock gate or the inactivation gate, it has closed. So now the channel is effectively closed, no sodium can get through. On the other hand, your potassium channel is open and potassium is able to leave the cell. Now technically when you get to back down to -55 millivolts, the sodium channel is going to change its state so that the regular gate is closed and the lock gate is open. But for most of the repolarization phase, it's in this locked state. And then finally we have our last phase, which is hyperpolarization. So in the hyperpolarization phase, basically your sodium channels are still closed. They're just regular closed, log gate open, but regular gate closed. And your potassium channels are still open, but they're working on closing. They were triggered to close back at -55 millivolts during the repolarization phase, but they're slow, so they didn't do it yet. And you got the membrane potential back down to -70 millivolts, which is rest. But your voltage-gated potassium channels were still working on it, so they're still not closed, which means that you still have potassium leaving the cell, which means your membrane potential is still going to be dropping. And it's actually going to go down below -70 millivolts. It's going to go all the way down to -90 millivolts, and that's your hyperpolarization phase. We call it the hyperpolarization phase because here your membrane potential is going back down below the resting value. Once you get to actually 90 millivolts, then your voltage-gated potassium channels will finally be ready to close. That is just, I guess, how far down you're able to go on the membrane potential while those channels are still working on closing, but before they actually close. So technically this phase should end whenever those potassium channels actually end up closing, and that's at about -90 millivolts. Then after that, the membrane potential is going to kind of rise back up slowly to the resting value. So I said that action potentials always happen the exact same way, and they basically do, but it's not actually every single neuron whose action potentials have this third phase, the hyperpolarization phase. Most of them do, like almost all of them do, but there's just a few examples of neurons that do not have the hyperpolarization phase. And that's because they have a different resting membrane potential. So the normal neuron has a resting membrane potential of -70 millivolts, but there's a few neurons that actually have a resting membrane potential of -90 millivolts, which means that basically the whole thing will happen the same way for them. And for them, just like for any other neuron, when you go back down, from being depolarized, you drop back down to -55 millivolts in repolarization, and your voltage-gated potassium channels are triggered to close, but they don't do it yet because they're slow. They don't actually close till you get to -90 millivolts. The only difference is that in these neurons, since the resting membrane potential is -90 millivolts, by the time you get back to -90 millivolts, you're just back at rest. You haven't actually hyperpolarized unless you go below the resting membrane potential. So that can only happen if your resting membrane potential is above -90, which it is for the majority of neurons, but not for a couple of them. So now I just have some figures kind of going through those stages of the action potential again. So here we have a section of the membrane which is in the axon hillock, and it is at rest. So your voltage-gated sodium channels are closed, lock gate open, regular gate closed. Your voltage-gated potassium channels are closed. And then you have local potentials coming in. So in green, you have these local potentials coming in. You're starting with the membrane at rest, but the local potentials will depolarize it, in this case taking it to threshold. So you can see that happening on this graph here, which is showing your membrane potential. So you start at rest, -70, then your local potentials cause the membrane potential to rise to -55 millivolts. Now we're going to trigger the action potential. So now that we're at -55 millivolts, now we can enter the depolarization phase. When we're entering the depolarization phase, our voltage-gated sodium channels will be triggered to open. They open at negative 55 millivolts. So that means now that both of their gates are open and sodium is able to rush into the cell down its concentration gradient, bringing positive charges into the cell, which will cause the inside of the cell to become more positive, and that means that the membrane potential will depolarize and start going up. Your voltage-gated potassium channels are still closed. And if we look at what's happening to the membrane potential, we see that indeed it is going up from negative 55, up, all the way to 30 millivolts. So this is what's going on in your depolarization phase. So after the depolarization phase, then we move to the repolarization phase. So at 30 millivolts, our peak depolarization value, we're going to have the sodium channels reset. So let's see. So these first two diagrams up here are related to the repolarization phase. In the first one, we see that our sodium channels have reset. So at 30 millivolts probably is where the lock gate is triggered to close. The regular gate is still open. The channel itself is effectively closed. No sodium can pass through. So that means we're done depolarizing the membrane now. Instead, what's happened is our voltage-gated potassium channels have opened. They were triggered to open at negative 55 millivolts, but they're slow, so they barely managed to do that. And now they're actually open, and potassium can leave the cell, flowing down its concentration gradient from where you have a lot of it to where you have a little of it, and taking its positive charges with it. So as these positive charges are leaving the cell, we have less positive charges left behind in the cell, and less positive means the same as more negative. So the membrane potential is going to be dropping. And we see that happening on the graph. The membrane potential starts to drop. It's actually going to continue to drop in the rest of the repolarization phase. These voltage-gated potassium channels will continue to be open for the rest of the repolarization phase. But once we get back down to negative 55 millivolts on our membrane potential, the sodium channels are going to reset. So their lock gate is going to open, and the regular gate is going to close back up again. The voltage-gated potassium channels will also be triggered to close at negative 55 millivolts, but they're slow, so they don't do it yet. So that's all happening at negative 55 millivolts, but the repolarization phase is going to continue until you get to negative 70 millivolts. Then once you get to negative 70 millivolts, the repolarization phase is over and the hyperpolarization phase begins. And that's shown at the bottom here. So at the beginning of the hyperpolarization phase, the membrane potential is continuing to drop below negative 70 millivolts, down actually to negative 90 millivolts, and that's because these voltage-gated potassium channels are still open. It's actually shown as closed here, but it should be shown as open for at least the first part of this phase. I guess depending on how you want to count the ending of the phase, which, it kind of doesn't really matter because the actual events that occur no matter what. But, in the first part of this phase, when your membrane potential is still dropping from negative 70 down to negative 90 millivolts, your voltage-gated potassium channels would still be open. That's the reason why the membrane potential is still dropping, because you still have potassium leaving the cell, taking it more negative. But of course, those channels were already triggered to close at negative 55 millivolts, so they will eventually actually close. And then your sodium channels will still be closed, and your potassium channels will be closed. And at that point, the membrane potential is going to stop dropping. That'll be at negative 90 millivolts, and then it'll start creeping back up to the resting value, which is actually kind of shown here. So you can kind of count the hyperpolarization phase as cutting off at negative 90 millivolts. That's probably the most appropriate place to count it as cut off when these channels finally get back to closed. But some people might consider that it continues at least until the membrane potential gets back to rest. Kind of depends on your preference. I won't ask you anything about that. Action potentials will also have something that we call the refractory period. The refractory period is the time after an action potential fires during which you cannot fire another one. And you have two kind of refractory periods for action potentials. First, you have the absolute refractory period, and second, you have the relative refractory period. During the absolute refractory period, that's kind of the first one that occurs. It's actually absolutely physically impossible to create another action potential. The absolute refractory period is going to be occurring during the depolarization and repolarization phase of your first action potential. So if you're during the depolarization phase, then you can't fire another action potential, because in order to fire an action potential, you have to open voltage-gated sodium channels and have depolarization. But you're already doing that for the first action potential, so you can't do it for the second one. If you open sodium channels, it's only going to contribute to this first action potential. It will not contribute to a second one until the depolarization phase is done. But once the depolarization phase is done, you go to the repolarization phase, and for most of that phase, the sodium channels are locked. Their lock gate has closed, but until you get back down to negative 55 millivolts. So as long as those sodium channels are locked, you cannot open them. So, in order to fire an action potential, you would need to take the membrane potential to above negative 55 millivolts and have those voltage-gated sodium channels open. But in the repolarization phase, through most of it, you are above negative 55 millivolts, but the sodium channel cannot open because it's locked. The lock gate is closed and the channel is locked. So it's impossible to make the channel open again until it resets. It has to open, lock, then reset back to closed. So during the absolute refractory period, it is impossible to make another action potential. In the depolarization phase, you're still busy depolarizing for the first action potential, and during the repolarization phase, at least for most of it, your sodium channels are locked and cannot physically open until they, reset and close regularly. After the absolute refractory period, you're going to enter the relative refractory period, which basically corresponds to the hyperpolarization phase. During the relative refractory period, it is possible to fire another action potential, but it's kind of hard. It's harder than normal to do it. You can still do it, but it is harder than normal. Which means that your regular local potential strength that would fire an action potential, that's not going to be strong enough anymore. Now the local potentials will have to be even more stronger to fire the action potential. So the reason why this is occurring is because your potassium channels are open during this time, and you have potassium actively leaving the cell, which is actively dropping the membrane potential. So if you're going to raise the membrane potential up to negative 55 millivolts, you're not going to have to just take it from negative 70 to negative 55. You're going to have to take it from maybe negative 70 to negative 55 in the face of a potassium current, which is trying to keep it down towards negative 70 or even below. Then once you get further into the hyperpolarization phase, your membrane potential is dropping even further. It's going down all the way to negative 90. So if you're trying to fire an action potential while that section of the membrane is at negative 90 millivolts, now you don't just have to take it from negative 70 to negative 55, you have to take it from negative 90 to negative 55, which requires a much stronger local potential. And not only that, but you still have your potassium currents leaving the cell and taking the membrane potential more negative. So you would still have to overcome those negative or hyperpolarizing potassium currents at the same time. It's sort of like if you have local potentials coming into the axon hillock, and most of them are depolarizing, but a couple of them are hyperpolarizing. Those hyperpolarizing ones are minusing you. They're taking you further away from threshold. So your depolarizing ones have to be even stronger to overcome those hyperpolarizing ones and then take it to threshold as well. In the hyperpolarization phase, your potassium currents are essentially hyperpolarizing. So you have to have depolarizing potentials that are strong enough to overcome that hyperpolarization from the potassium. And you can do that, but it's just hard. It's just hard to do it. So here you have kind of a graphical representation of the action potential with the relative and absolute refractory periods. So first we'll just look at the action potential itself. You're starting at negative 70 millivolts at rest. Then you have some depolarizing local potentials coming in, raising your membrane potential up to negative 55 millivolts. Now your sodium channels are going to open. So once your sodium channels open, sodium will rush into the cell. that takes the membrane potential high up all the way to 30 millivolts. At that point, your sodium channels are going to lock, and your potassium channels will finally be open. So now you have potassium leaving the cell. That is going to take your membrane potential all the way back down, to rest, actually, negative 70 millivolts, so that's the repolarization phase, but actually at negative 55 millivolts, those sodium channels will reset and go back to just being regular closed. Then, once you're at negative 70 millivolts, you enter the hyperpolarization phase, and the membrane potential continues to drop because you still have potassium leaving the cell. Only when you get down to like negative 90 millivolts do those potassium channels actually close. So once you get down to negative 90 millivolts, your potassium channels close, and your membrane potential can start to slowly move back up to the resting value of negative 70 millivolts. So you could call the end of the hyperpolarization phase right here at negative 90. Or you could call it, I guess, when you actually get back to your resting membrane potential. In blue here, in light blue, I guess it's all in blue, but in light blue you have your absolute refractory period shaded in, and in darker blue you have your relative refractory period shaded in. So with the absolute refractory period first, in the depolarization phase, you can't fire another action potential because you're already firing the first one. You're not done with that yet. All these sodium channels that are open are contributing to the first action potential. They will not contribute to a second one. So it's impossible to make another action potential during this time. Then you have, during your repolarization phase, during most of it actually, you have the second half kind of your absolute refractory period. So here, your sodium channels are just locked. The lock gate is closed on them. They will not open until you reset them by making them go back to kind of the regular closed state, which happens at negative 55 millivolts. So that's the reason why the absolute refractory period will end at negative 55 millivolts, which is almost at the end of the repolarization phase, but like not quite. So during this whole time, it's impossible to make another action potential. Then, once we get into this darker blue shading, now it is technically possible to make another action potential, but it's just harder to do it than normal because we are in the relative refractory period. So what's happening here is that our sodium channels have reset. So now they're just in the closed state, which means that actually it is possible to make them open again. You would have to make the membrane potential go back to negative 55 millivolts. And for most of this phase, like, I guess at the very, very beginning of it, it's basically at negative 55 millivolts. But, from that point on, the membrane potential is continuing to drop as potassium is continuing to leave the cell. So it's going to be really hard to make that gate open. That voltage gated sodium channel, to make that open, because you have to get all the way back to negative 55 millivolts. And through part of this phase, you are like lower than negative 70, so it's harder than regular because of that. And for all of this phase, you have potassium leaving the cell. And so that potassium is taking you negative. If you're trying to go positive, you have to it's like lifting a box while someone else is like pushing down on the top of the box. It's going to be hard. You could maybe do it if you're strong, but it's hard. So now we'll talk about how an action potential moves along an axon, or that process is called action potential propagation. So when you have your axon hillock, which is the very base of the axon, depolarizing for the depolarization phase of an action potential, you have all this sodium rushing into the axon at that point. And that sodium isn't going to just stay put, but it's going to start diffusing out along the membrane. So it'll end up diffusing down the axon to kind of the next portion of that axon membrane that is like right adjacent to the axon hillock, and will actually depolarize the membrane at that point. And it actually ends up being enough to depolarize the membrane to threshold. So at the axon hillock, where the sodium is actually entering through these voltage-gated sodium channels, you have depolarization all the way up to 30 millivolts. And in the kind of adjacent portion of the axon membrane, where you don't have any open sodium channels yet, you just have sodium ions diffusing there, you're going to have not as much depolarization, but still enough depolarization to take that part of the membrane up to threshold of negative 55 millivolts. Once that adjacent portion of the membrane reaches negative 55 millivolts, that is going to trigger the voltage-gated sodium channels in that part of the membrane to open. So once those channels open, you're going to have sodium rushing into the axon at that point, and that part of the axon then will enter the depolarization phase of an action potential. Roughly at about the same time that your adjacent part of the membrane is entering the depolarization phase, the first part of the membrane at the axon hillock is going to be entering the repolarization phase. It'll be in the absolute refractory period for the majority of that phase. So the membrane potential will still be above negative 55 millivolts, and the sodium channels will be inactivated or locked, which means that it's impossible for that part of the membrane to enter an action potential again. You're still during that absolute refractory period when it's impossible to start another action potential. So when these sodium ions start rushing into the axon in your adjacent part of the membrane, they're going to also be diffusing forwards and backwards along the axon's membrane. So they will diffuse backwards to the axon hillock, but they're not going to be able to start another action potential there because you're still in the absolute refractory period. When they diffuse forward, though, to the next part of the axon membrane, they actually will raise that part of the membrane up to threshold of negative 55 millivolts and start an action potential there by opening those voltage-gated sodium channels. And in this way, the action potential is just going to kind of move down the membrane, down the axon, from one part of the membrane to the next part of the membrane that's adjacent to it. And it'll only go in the forward direction towards the end of the axon. It'll never go backwards, back towards the beginning of the axon, because the part of the membrane that it would have to travel through to go back to the beginning of the axon is always going to be in the absolute refractory period. So here we just have a diagram kind of showing that process. So here you have the membrane of an axon, or just an axon with, the membrane on either side of it. And you have your voltage-gated channels in it, purple for sodium and blue for potassium. This first part of the membrane is the axon hillock, or the trigger zone, and then beyond that you have the rest of the axon. So in this first image here, we have local potentials arriving at the axon hillock and adding up together. So all these sodium ions here are sodium ions that are, underlying a local potential that is arriving at the axon hillock. So the membrane is then going to depolarize to threshold. And when that happens, you're going to have these voltage-gated sodium channels opening. And sodium ions will rush into the cell and they will begin diffusing. They're going to diffuse in both directions, forward, down along the axon, and also back towards the cell body. They're only shown diffusing in one direction because that's the only direction that kind of matters, but they actually will diffuse backwards into the cell body as well. As they're diffusing backwards, it doesn't impact anything because there are no voltage-gated channels over this way, so you're not going to be able to start a action potential if you're going back to the cell body. But these sodium ions that are diffusing down towards the rest of the axon membrane, you do have the voltage-gated channels in that part of the membrane, so as those sodium ions diffuse, they are changing the membrane potential, and when the membrane potential at this adjacent portion reaches negative 55 millivolts, then these voltage-gated sodium channels are going to open. And that is what is going on right here. So here you've had sodium ions diffuse from this first channel down the axon, and they depolarize the membrane in this adjacent section of the axon to threshold, and these voltage-gated sodium channels in this part of the membrane opened. So now you have sodium rushing in. It's going to diffuse again into the cell in both directions. So it'll be traveling back down the axon back up the axon, I guess I should say, towards the cell body. It'll also be diffusing down towards the rest of the axon. But when these sodium ions are traveling back towards the cell body, they're going to first reach this adjacent part of the axon, which is kind of, in this case, the first part of the axon, and this part of the axon is now in the repolarization phase of its action potential. So you have your voltage-gated potassium channels open, potassium is leaving the cell, and it is actually impossible to fire a new action potential because your voltage-gated sodium channels are locked or inactivated. So it's impossible to open them back up until they reset. So all this sodium that's diffusing here isn't going to impact anything. The sodium that's diffusing down this way towards the rest of the axon is going to have an impact, though. As it travels, it is raising the membrane potential, and when it reaches this next kind of portion of the axon's membrane, it will raise the membrane potential enough for it to reach threshold of negative 55 millivolts, and then these voltage-gated sodium channels will open. When that happens, sodium will start rushing into the cell through those channels. It'll again diffuse down the membrane and back up towards the cell body. But again, when it starts diffusing back up, it doesn't do anything because it's just hitting this part of the membrane where you have voltage-gated sodium channels that are inactivated or locked, because this part of the membrane is now in the repolarization phase of its action potential. So the action potential is just going to stop, or not stop, but continue moving down the axon, one section of membrane at a time until it gets to the end. So there's actually two types of action potential propagation, or two kind of ways that the action potential can move down an axon. And the way that I just talked about is the regular way, or that's what occurs when you don't have myelin around your axon. So that's called continuous propagation, because you're just moving from one section of membrane to the adjacent section of membrane. But if you have an axon that is wrapped in myelin sheaths, then you're going to have a different type of action potential propagation, which is called saltatory propagation. Saltatory just means jumping. So the action potential is going to be jumping between the myelin sheaths from node to node, or from one node of Ranvier to the next one. So here you have kind of a graphical comparison of those two types of action potential propagation. So on the top you have the saltatory action potential propagation, and on the bottom you have continuous action potential propagation. So on the bottom, your action potential is just moving from one segment of membrane to the adjacent segment, to the adjacent segment, and to the adjacent segment, so on and so forth, until it gets to the end. On the top you have a myelinated axon. So when your action potential begins and at the axon hillock, and you have sodium ions rushing into the cell there, they're actually going to diffuse all the way across this, or through this myelin sheathed area of the axon, to the next node of Ranvier. The next voltage-gated sodium channel that they're actually able to open is going to be at this next node of Ranvier, because where you have the myelin sheath, no ions are able to cross the membrane at that point. So you're not going to be able to do anything until you get to the next node of Ranvier. So those sodium ions are going to diffuse down the axon to the next node of Ranvier. They're going to open the voltage-gated sodium channels that you have there, and then that part of the membrane will enter the depolarization phase of an action potential. Its sodium ions, or the sodium ions that are coming in now here at this part of the membrane, are going to diffuse actually again in both directions along the axon. But when they go backwards, they're going to get to part of the membrane that is in the absolute refractory period, because it's in the repolarization phase and the sodium channels are locked, so they can't be reopened. Only when the sodium ions diffuse down the axon will they get to a new part of the membrane, or the next node of Ranvier, where it's basically at rest already, and so those sodium channels are ready to open as soon as the membrane is depolarized to threshold. So those sodium ions are going to depolarize this part of the membrane to threshold, and then the next set of voltage-gated sodium channels will open, and that part of the membrane will enter the depolarization phase of an action potential. So the action potential is just going to hop from node to node to node, and that is ending up going much faster than this continuous action potential propagation that isn't skipping any parts of the membrane and is just depolarizing the very next part of the membrane each time. So kind of the main thing that affects the speed at which axons conduct action potentials is just going to be whether they're myelinated or not. Also, sometimes you might have really thick myelin that kind of works better as insulation versus really thin myelin, and that doesn't really work as well. But you also have one more factor that affects how fast axons can conduct action potentials, and that's just the diameter of the axon itself. So in all cases, the fatter the axon is, or the bigger its diameter is, the faster it's going to be able to conduct action potentials. So part of that has to do with the resistance and capacitance of the axon itself, depending on its size. We're not going to really worry about that because in order to understand that, you have to understand like physics, and it's just beyond our purview. But there are kind of physical laws about electricity that apply to axons and to cells and to membranes that are affecting the speed at which different sizes of axons can conduct action potentials. But apart from that, you just have the simple fact that a larger axon with a bigger diameter is going to have more membrane wrapped around it, just a larger surface area of the membrane. And when you have a larger membrane, you can have more voltage-gated channels embedded in it. And if you have more voltage-gated channels, then you're going to have more channels that open during the depolarization phase of the action potential, and more sodium is going to be able to enter the axon during that time period. Then, since you have more sodium, it's going to diffuse faster to the next part of the axon membrane or to the next node of Ranvier, which means that it's going to be able to depolarize that next part of the membrane or that next node more quickly. And then that part will enter the depolarization phase of an action potential more quickly. When you have a larger axon, like most of it is still going to be filled up with cytoskeleton. So in all cases, for all axons, most of the volume inside the axon is just occupied by the cytoskeleton going through it. So when you have a larger axon, you effectively don't have much more free space inside of it. You just have more cytoskeleton. So when the sodium enters the axon, it's going to start diffusing mostly just along the membrane there. It won't diffuse as much like kind of down into the interior of the axon because that space is already taken up by the cytoskeleton. So it'll just stay kind of on the surface. Then if you have more of it, it's going to be able to diffuse faster along that surface. So that means that out of all the axons in your body, the fastest ones that are there are both large in their diameter and thickly myelinated. The slowest axons in your body are really small in their diameter, and they are not myelinated at all. And then I'll just kind of note that the textbook goes through different types of axons, or kind of different categories of axons. Like different axons are categorized basically on their speed, which also means kind of on their myelination and on their size. But that is only applying to the peripheral nervous system. So that doesn't apply to any axons that are in the brain or the spinal cord, only to axons in the nerves. And you actually don't need to know those categories of axons. So this diagram is kind of just summarizing everything that we just talked about related to local potentials and action potentials. So we're seeing a neuron here, and you also have another neuron that's contacting it, or maybe a couple of neurons. We're seeing a couple of axons coming in here. Maybe those are both branches of the same axon, or maybe they're actually different, different axons from different neurons. But in any case, maybe we'll assume that they're two different axons from two different neurons. So in this case, or in that case, these are going to be your presynaptic neurons, and then this neuron here is your postsynaptic neuron, when we're talking about these synapses here that are formed between the two. So these axons are coming in, branching into telodendria, and then you have the axon terminals at the very ends. Each of those axon terminals is forming a synapse with part of this postsynaptic neuron. Most of them are on the dendrites, but you can see one of them here is actually on the cell body itself. Another one being at the very base of the dendrite. So you're going to have action potentials coming in along these axons, arriving at these synapses, and when they get to the synapses, they're going to trigger them to release neurotransmitter. That neurotransmitter is going to cross the synaptic clefts and go to the postsynaptic cell, where it's going to bind its receptors. Those receptors are most likely an ion channel, a ligand-gated ion channel. So when you have the neurotransmitter bind to this ligand-gated ion channel, the channel is going to open and allow an ion to flow through. And in this case, we're going to assume that these are sodium channels, so you have sodium ions entering the cell at these synapses. those sodium ions are going to create local potentials that are depolarizing, because the sodium is always going to take the membrane to a more positive potential, so it's depolarizing. Then these kind of blue lines here are showing the local potentials traveling down the dendrites and across the cell body to the axon hillock. Once they get to the axon hillock, they're all going to kind of merge together and add up. And in this case, there's enough of them that even though they've decayed as they traveled, they still add up to enough at the axon hillock to take the membrane potential from -70 at rest up to -55, which is your threshold potential for triggering an action potential. So once the membrane here reaches -55 millivolts, you're going to have voltage-gated sodium channels here open, and you see sodium entering the cell. That sodium is going to start diffusing along the axon. It's actually going to diffuse maybe to here and open some more voltage-gated sodium channels, and then you'll have more sodium entering the cell. That'll diffuse across this myelin sheath to this first node of Ranvier, and it'll end up depolarizing this part of the membrane to threshold. And you're going to have more voltage-gated sodium channels opening here and more sodium entering at this point. So at that point, you're going to have an action potential that started here and spread to this node of Ranvier. Then it's going to end up hopping to this one, and this one, and this one, and this one, and so on and so forth. But as it's just reached this first node of Ranvier, so as this first node is entering its depolarization phase, this first part of the axon's membrane is going to be in the repolarization phase. Which means that it's in the absolute refractory period for most of that phase. The sodium channels are going to be locked. The potassium channels will be open, but the sodium channels are locked, and you cannot start another action potential before you reset those channels. So it's impossible to fire another action potential, so we say that this part of the membrane is in the absolute refractory period. So that means that as sodium ions are entering the membrane at this point, they're going to be diffusing in both directions. They will diffuse back to this first part of the membrane, but they're not going to start an action potential because it's in the absolute refractory period. As they diffuse to the next node of Ranvier, though, they will start an action potential there, and this part of the membrane will enter the depolarization phase. As this part of the membrane is kind of in the depolarization phase for its action potential, this part will be in the repolarization phase or in the absolute refractory period. And this part will likely have entered the hyperpolarization phase of its action potential and just be finishing that up, which means that it will be in the relative refractory period, where it is possible to fire a new action potential, but it's just harder to do it than normal because the membrane is going to be hyperpolarizing down to -90 millivolts, so you just have to take the membrane potential a farther distance to get up to -55 if you start at -90 versus at -70. And also you're having active potassium currents leaving the cell. So that's also just making it harder to change the membrane potential in a positive direction. But you technically can do it if you had really a lot of strong local potentials arriving at the axon hillock at the same time. Then it would be enough to do it. So this action potential is going to travel along down the membrane. And I guess down here we're actually showing that saltatory conduction where you have sodium ions entering at one node of Ranvier, diffusing down to the next node, depolarizing it, opening your voltage-gated sodium channels at that part of the node, or that part of the membrane, or at that node. More sodium ions rushing in, diffusing down the membrane, opening voltage-gated sodium channels at the next node of Ranvier, so on and so forth, until eventually the action potential actually reaches the end of its axon and goes down these telodendria to the axon terminals, and is going to activate those terminals and cause them to release neurotransmitter. And that process of how the action potential causes neurotransmitter to be released and what all is going on at the synapse is kind of what we're going to talk about next. Okay, so we'll go over the synapse and how activity occurs at the synapse. Then we'll get into the neurotransmitters themselves, and then we'll be done. So first, just reviewing the basic anatomy of a synapse. You always have a presynaptic neuron that is sending messages and a postsynaptic neuron that is receiving those messages. So the presynaptic neuron is going to have an action potential travel down its axon that reaches the axon terminals at the synapse. Then it's going to activate that synapse and cause neurotransmitter to be released. That neurotransmitter will cross the synapse, crossing the synaptic cleft, which is the little space between the axon terminal and the dendrite of the postsynaptic neuron. And when it gets to the other side, it'll bind to its receptor, causing a local potential to begin in this postsynaptic cell, which means that the message is basically received. So in this blown up view, you can see the neurotransmitter being released from the axon terminal, crossing the synaptic cleft and binding to its receptors. These neurotransmitter receptors are probably ligand-gated ion channels that are going to open when the neurotransmitter binds, and then ions will cross the membrane, and that will cause a local potential to occur. This neurotransmitter, after it has opened those ligand-gated ion channels that are its receptors, it's going to kind of let go of its receptors, and then it's going to be pumped back into the presynaptic cell's axon terminal by a neurotransmitter transporter molecule that is just recycling it. Okay, so here is a more detailed kind of overview of what's actually occurring at that synapse when the action potential arrives. So first you have your action potential coming into the axon terminal. That means that you're going to have this part of the axon's membrane entering the depolarization phase of the action potential. As the action potential is arriving, that just means that this part of the membrane is entering the depolarization phase. So you're going to have a lot of sodium coming in here, and that's going to end up depolarizing the membrane of the axon terminal. And in the axon terminal, you have special voltage-gated channels. So you don't have the voltage-gated sodium or potassium channels that we're used to seeing. Instead, you have these voltage-gated calcium channels. These calcium channels are going to open when the action potential arrives and depolarizes the axon terminal. And then calcium is going to enter the cell. There's more calcium outside of the cell and less calcium inside of the cell. So calcium will enter the cell down its concentration gradient through these open channels. That is going to end up depolarizing the axon terminal's membrane, but that's not actually the most important thing that's going to happen there. The really important thing is that calcium is actually going to bind to these synaptic vesicles that are storing the neurotransmitter and cause them to fuse with the axon terminal's membrane. So the neurotransmitter isn't going to be just kind of floating around in the axon terminal freely. It's actually going to be packaged up into these little membrane sacs that are called vesicles. In this case, they're called synaptic vesicles because you find them at the synapse and they're storing neurotransmitter to be released at that synapse. And it's basically just going to keep that neurotransmitter safely stored away and just wait. And is waiting for calcium to come and bind to some of the proteins that are attached to this vesicle. So calcium is actually going to bind to proteins on this vesicle, and when that happens, those proteins will be activated and they will cause the vesicle to move towards the membrane of the axon terminal where it faces the synaptic cleft, and then fuse with that membrane. When the vesicle's membrane fuses with the axon terminal's membrane, you end up having the contents of the vesicle released into the synaptic cleft. So these neurotransmitters are going to be released because of calcium that entered the axon terminal through voltage-gated calcium channels that opened when the action potential arrived. Then the neurotransmitter is going to cross the synaptic cleft and bind to its receptors, which in this case are ligand-gated ion channels. When neurotransmitter binds to those receptors, the channels will open and ions will start flowing through. In this case, these are sodium channels, so we have sodium ions that are going to be entering the postsynaptic cell now and depolarizing it, which causes a depolarizing local potential. Then the neurotransmitters themselves are going to be cleaned up. How they're cleaned up exactly kind of actually depends on which neurotransmitter we're talking about. Most of them will be cleaned up by being pumped back into the presynaptic cell by a neurotransmitter transporter, which is actually not shown in this diagram. But there are some neurotransmitters that are not cleaned up in that way. Instead, you actually have enzymes in the synaptic cleft that degrade them. So there's lots of different types of local potentials that can be elicited or caused by neurotransmitters binding to their receptors at a synapse. So some of those local potentials will be depolarizing, which means that they're excitatory because they take you closer to an action potential. Some of them will be hyperpolarizing, which means that they're inhibitory because they take you farther away from firing an action potential. Some of them will be small, so it'll be just kind of a small change to the membrane potential. Some of them will be a bigger change to the membrane potential. So it kind of depends on how much neurotransmitter you're releasing and on what specific ion channels are going to be opened in response. So if you're releasing a small amount of neurotransmitter at the synapse, you're going to have a small local potential result from that. You're going to only be able to open those ion channels for probably a small amount of time, or maybe you will only be able to open like a small subset of the total number of ligand-gated ion channels that are present at the synapse. So either way, you're going to have just a few channels open for a little bit of time, and you're only going to get a little bit of ions crossing through those channels when they're open, which gives you a small local potential. On the other hand, if you have a large amount of neurotransmitter released at this synapse, then you're going to have your ligand-gated ion channels being open for a longer period of time, and you're going to have a greater number of them being opened, which means you're going to get a lot of ion crossing the membrane and a large local potential. So the kind of magnitude of the local potential or the size of the local potential is going to mostly depend on how much neurotransmitter is released at the synapse. Then the direction of the local potential, so whether it's depolarizing or hyperpolarizing, whether it's excitatory or inhibitory, that is going to depend just on what ion or ions the ion channel that opens is able to allow to cross the membrane. So you actually have a lot of channels that only allow one ion to cross, but you have other channels that will allow two or more ions to cross at the same time. So what type of local potential you get just depends on which ions are crossing. Anytime that you have chloride crossing or potassium crossing, that means that it's going to be a hyperpolarizing local potential, which means it'll be inhibitory. Anytime you have sodium ions crossing, that's going to be a depolarizing local potential, which is excitatory. So when sodium crosses, it brings positive charges into the cell that causes depolarization, which is excitatory. If you have chloride crossing, it's going to be negative charges coming into the cell because you have more chloride outside the cell and less inside the cell. So chloride always goes into the cell through a channel if it's open. So that means that negative charges are entering the cell. That's going to take the membrane potential more negative, which is hyperpolarizing, which is inhibitory. For potassium, you have more inside the cell and less outside. So when potassium channels open, potassium will always flow out of the cell. That means that you have positive charges leaving the cell. So you have less positive charges kind of left behind in the cell, and less positive means the same as more negative. So when potassium leaves the cell, it's always going to take the membrane potential more negative, which is hyperpolarizing, which is also inhibitory. So here's just a couple more diagrams of that, where you have a synapse, activity at the synapse, so an action potential arriving at the synapse, causing neurotransmitter to be released, and then you can see all these little tiny dots here on the postsynaptic neuron. Those are actually ligand-gated ion channels that are receptors for that neurotransmitter. So on the left-hand side, those are sodium channels. So when they open, sodium enters the cell, positive charges enter the cell, it is depolarizing, and you can see the depolarizing local potential on the graph as the membrane potential goes more positive. On the right-hand side, you have chloride channels instead. So when those channels open, chloride is going to be entering the cell, and that's a negative charge. So the membrane potential will drop down to be more negative, and that's shown in this graph here. I guess I'll mention that another name for a depolarizing local potential is an EPSP, which means excitatory postsynaptic potential. And another name for a hyperpolarizing local potential is an IPSP, which stands for inhibitory postsynaptic potential. I'm not really going to use that terminology because I just feel like it's simpler and more direct to say depolarizing and hyperpolarizing local potential. But if you run across it, EPSP is just a depolarizing or excitatory local potential, and IPSP is a hyperpolarizing or inhibiting local potential. Okay, so that's basically what is going on at the synapse. Now we'll get into the neurotransmitters themselves and the phenomenon of neuromodulation, which many neurotransmitters are involved with. So, neuromodulation is kind of other ways that neurotransmitters can affect the postsynaptic cell apart from just causing local potentials to occur in that cell. So far what we've talked about is that you have activity at a synapse, which means an action potential is arriving at that synapse. Then you're going to have depolarization of the axon terminal, voltage-gated calcium channels open. Calcium enters the cell, it binds to proteins on the synaptic vesicles that are storing the neurotransmitters. It causes those vesicles to fuse with the axon terminal membrane where it faces the synaptic cleft. That causes those neurotransmitters to be released into the synaptic cleft. They bind to the receptors, which are a ligand-gated ion channel. They open those ion channels. Then you have ions crossing the membrane, and that causes a local potential. Actually, some neurotransmitters do not use a ligand-gated ion channel receptor. Sometimes they use a different type of receptor, which is called the G-protein coupled receptor, which we'll talk about in a moment. But those neurotransmitters that are using that other type of receptor, they're not going to be causing they're not going to directly cause local potentials to occur because this type of receptor is not an ion channel. So when they bind to the receptor, something else is going to happen apart from just an ion channel opening. That something else is going to be basically changes in how proteins inside the postsynaptic cell are functioning. So you're going to have like some of those proteins turning on, some of them turning off. You can actually have changes occurring in the gene expression for that postsynaptic cell as well. So some of these proteins that turn on may end up turning on certain genes or turning off certain genes so that you would make the protein that is associated with that gene, or you would stop making the protein associated with that gene. So in neuromodulation, you are altering kind of the protein function of the postsynaptic cell, or you're altering the gene expression of the postsynaptic cell. And that can have really far-ranging effects on that cell in terms of changing how it functions and even changing its basic structure. Ultimately, these changes are going to change how the synapses are functioning. Maybe it'll change how the postsynaptic neuron is responding to neurotransmitters in the future. Maybe it'll change how the presynaptic neuron is actually releasing neurotransmitters in the future. So we call it neuromodulation because you are modulating how the synapse is working, either by changing how the postsynaptic cell responds or by changing how the presynaptic cell releases neurotransmitter. This is a basic type of neuroplasticity. So when the nervous system is changing, it's always going to be involving changes in protein function or changes in gene expression. Some of those changes might be kind of minor or might not last for a long time. Some of them might be really major changes that last for a very long time. You can actually have new synapses being built this way. That would be an example of a really large change that can occur through neuromodulation. So you have to have those types of changes in order for learning and memory to occur. In order for you to learn something, you have to actually change the structure of your nervous system. And the way to change that structure is through neuromodulation, by either changing protein function in your neurons or changing gene expression in those neurons. So in addition to neuromodulation actually creating new synapses, it can also just kind of strengthen existing synapses. When the synapse is strengthened, basically what that means is that when you release a little bit of or I guess I should say, when you have a small amount of action potentials arriving at that synapse to activate it, you're going to either release a lot more neurotransmitter than normal, or you're going to release the regular amount, but then the postsynaptic cell is going to respond to it really strongly. Either way, you're going to get stronger or more and stronger local potentials in the postsynaptic cell in response to less number of action potentials in the presynaptic cell. So that means just if you're sending a weak signal, it gets received as a strong signal if the synapse is strengthened. But neuromodulation can also weaken synapses, which is basically the opposite thing, where even if you're sending a really strong signal at the presynaptic neuron with a lot of action potentials, you're only going to get a weak signal received by the postsynaptic neuron. So you're going to end up with weak local potentials, either because you didn't release a lot of neurotransmitter or because you didn't respond to the neurotransmitter very much. So neuromodulation usually involves G protein coupled receptors instead of ligand-gated ion channel receptors. G protein coupled receptors are also called metabotropic receptors, and they are not an ion channel. Instead, it's just a protein receptor that is coupled to another protein that is called the G protein. So when you have your neurotransmitter binding the G protein coupled receptor, the G protein part is going to kind of let go of it and move off and start doing things inside the cell. And it's actually going to kick off a signaling cascade inside the cell, or just a chain of events, a chain of, one protein contacting another protein and turning it on, that protein turns on another protein, that protein turns on another protein, a bunch of different enzymes activating. And at the end of the signal cascade, you're going to get some type of functional change in the cell. So actually sometimes when you have G protein coupled receptors activated, the G protein is going to kick off the signal cascade that ends up opening ion channels. So G protein coupled receptors can actually end up causing local potentials indirectly if they lead to the activation of an ion channel. Those ion channels might be for sodium, for chloride, for potassium, but actually sometimes they're for calcium too. If we have a calcium ion channel opening, then we might get a local potential or we might not get a local potential, but we will get another signal cascade activating because calcium is a signaling molecule inside the cell. So it's not just having effects on membrane potential as an ion, but it actually will bind to proteins and turn them on or turn them off. So some other things that can happen when you have a neurotransmitter binding to its G protein coupled receptor is that you could have different proteins in the cell turning on or turning off. So you can have alteration in the protein function of the cell, or you can have alteration in the gene expression. So you could have some genes turn on and start making their protein product, or you could have other genes turn off and stop making their protein product. It could be any combination of those things actually. So when you have one G protein coupled receptor activated, you might end up having multiple functional changes in the cell. Maybe you'll have changes in protein expression and changes in gene expression. Maybe you'll have a local potential, you'll have another ion channel open that causes a local potential, and you'll have changes in protein function. Could be any combination of those things. And actually, any given neurotransmitter is probably going to have multiple types of G protein coupled receptors that it can bind that would be found in different parts of the nervous system. And all of those different receptors can end up doing different things. So, for instance, for serotonin, it has a lot of different G protein coupled receptors. I actually don't remember how many off the top of my head, but like at least 5. So each one of those receptors is going to do somewhat different things. They may have overlap in the kind of results that they cause, or but they're also going to do some extra things that are different from each other. And also, you might even have for one neurotransmitter that's being released at a synapse, you might have multiple different types of receptor for it at that synapse. So you might have one G protein coupled receptor there, and you might have another ligand-gated ion channel receptor there. It just depends on the synapse. So here you have a couple of diagrams of G protein coupled receptors. So this is the G protein coupled receptor in blue, and then next to it is the G protein itself. So the little black dot here is the neurotransmitter that is binding to the G protein coupled receptor that is going to activate this G protein. When that happens, this alpha subunit of the G protein is going to dissociate from it and float off inside the cell to do something else. In this case, it is floating over to an ion channel, a ligand-gated ion channel that uses this alpha subunit as its ligand and is going to activate that channel and open it. And in this case, it looks like we have sodium ions entering the cell, so that would cause a depolarizing local potential. On the other hand, on the right-hand side, we have something more complicated occurring. So we have our G protein coupled receptor again. The neurotransmitter binds it. That's going to activate the G protein that it is coupled to. When that protein activates, the alpha subunit is going to float away from it. In this case, it's going over to this enzyme and activating this enzyme. When this enzyme is activated, it's going to create this second messenger molecule, which is basically going to take the message that the cell is being activated, down into the cell. It's going to end up turning on other proteins, turning off other proteins, maybe changing gene expression. But another thing it's actually doing is opening this ion channel to also allow sodium to enter the cell and cause a depolarizing local potential. So this particular G protein coupled receptor is doing multiple things. It is indirectly opening an ion channel and indirectly doing some other stuff inside the cell that will probably affect protein function or gene expression in that cell for neuromodulation. So I had mentioned that neuromodulation can change the way that the postsynaptic cell responds to neurotransmitter, but it can also change the way that the presynaptic cell releases neurotransmitter. But so far we've only talked about neurotransmitters crossing a synapse, binding to the receptors, and then that changing something inside that postsynaptic cell. So that can change how the postsynaptic cell is going to respond to neurotransmitter, but that's not going to be able to change how the presynaptic cell releases neurotransmitter. So how could neuromodulation change how the presynaptic cell releases neurotransmitter? Well, there's a couple of different ways. One way is by having neurotransmitter receptors on the presynaptic cell itself. And that's what's shown in this diagram here. So here you have a cell that is releasing dopamine, norepinephrine, and serotonin as its neurotransmitters. So you have receptors on the postsynaptic cell for each of those neurotransmitters. And then we also have some transporters here for each of those neurotransmitters to recycle them back into the presynaptic cell once they're done signaling. But on the other side, in green, we have these autoreceptors. So these are basically just receptors for those neurotransmitters that are located on the presynaptic cell. So let's say for dopamine, when that neurotransmitter gets released, some of it is going to bind to the dopamine receptors on the postsynaptic cell. Some of it is actually going to diffuse to the side of the synapse and actually a little bit out of the synapse, and it'll end up binding to this dopamine autoreceptor that is located on the presynaptic cell. So at that point, this is probably a G protein coupled receptor, and you're going to have some changes occurring in this presynaptic cell that may affect how it is releasing neurotransmitter in the future. Some changes to its protein function or to its gene expression that change how it releases dopamine in the future. So that's one way that neuromodulation can affect the presynaptic cell. Another way is just based on where synapses are actually located. So the kind of standard synapse that we talk about is between an axon and a dendrite. But you also actually have other types of synapses. You can have the axon of one cell contacting the cell body of another cell directly. And you can also have the axon of one cell contacting the axon of another neuron, which is called an axoaxonic synapse. So in this case, when this cell is having action potentials travel down its axon, it's actually going to be having a neurotransmitter released onto the axon of the postsynaptic cell, which can cause changes in how that cell's axon is functioning. So it wouldn't be affecting how this synapse functions, but how these synapses function. And this is kind of a zoomed in view of that. So here you have one presynaptic cell, which would be this presynaptic cell. If you have action potentials coming down this axon, it's going to release these green neurotransmitters. And you have receptors for those neurotransmitters, which are metabotropic or G protein coupled receptors on the axon terminal for the postsynaptic cell, which I guess is, so the specific synapse is drawn in a slightly different location, but onto the axon or the axon terminal of your postsynaptic cell. And that postsynaptic cell is itself involved in another synapse, for which is the presynaptic cell. So that cell is going to be also releasing these red neurotransmitters as the presynaptic cell onto the postsynaptic cell right here, which in this diagram is a cell that's not shown. So this first presynaptic cell with the green neurotransmitters can release these neurotransmitters. They can bind to G protein coupled receptors on your postsynaptic cell, and those receptors can end up kicking off a signaling cascade that is going to alter the way that this postsynaptic cell is then sending messages using its synapse, for which is the presynaptic cell. So it can alter the protein function of the cell and alter the gene expression of the cell to change the way that these red neurotransmitters are being released in the future. Then finally, I had already kind of mentioned the idea of the tripartite synapse, or the idea that you have astrocytes also participating in synapses, astrocytes that have their own neurotransmitters and their own neurotransmitter receptors. So when this is happening, the astrocyte is basically going to be just involved in neuromodulation. So you're not going to have any ligand-gated ion channel receptors on the astrocyte. The astrocyte doesn't carry action potentials or anything like that. And in general, it's not going to be releasing neurotransmitters that cause action potentials in your postsynaptic cell. Instead, it's going to be releasing neuromodulators, or neurotransmitters that are going to bind to some G protein coupled receptor in the post or the presynaptic cell and cause changes in the protein function or the gene expression of that cell that will affect how it releases neurotransmitter or how it responds to neurotransmitter. The receptors that are actually on the astrocyte are also probably going to be G protein coupled receptors that are changing the way that the astrocyte functions in the synapse. And now that we've gone over neuromodulation, we can get into the individual neurotransmitters and kind of like the basic categories of neurotransmitters, which is the last major thing that we will talk about in this chapter. So you've got 4 basic types of neurotransmitters. First, you have your classical small molecule neurotransmitters. They're called classical because they're kind of the first ones that were discovered, and small molecule because, well, they are small molecules. These are usually not neuromodulatory transmitters, so usually they're going to be causing local potentials directly using a ligand-gated ion channel receptor, either be causing depolarizing local potentials or hyperpolarizing ones. They can also do neuromodulation sometimes, it's just a little less common for them. Then you have the neuromodulatory classical small molecule neurotransmitters, which are, again, classical because they're some of the first that were ever discovered, and also small molecule because they're small molecules. But these ones are neuromodulatory, which means that their primary role in the nervous system is to cause neuromodulation, to alter the way that synapses function in terms of how neurotransmitter is released or how it is responded to. So they can also evoke local potentials in some cases, but they're more focused on neuromodulation. Then you have the neuropeptides, which are just neurotransmitters that are made out of amino acids, short chains of amino acids that we call peptides. These are also going to be almost always neuromodulatory, so usually not causing local potentials, usually causing neuromodulation or altering the way that cells release or respond to neurotransmitter by changing protein function or gene expression in those cells. And then finally, the last type of neurotransmitter is the retrograde neurotransmitter, which is also neuromodulatory. So retrograde neurotransmitters are called retrograde because they travel backwards. So retrograde just means like going backwards. So the retrograde neurotransmitters are actually released from the postsynaptic neuron and they travel back to the presynaptic neuron. And the presynaptic neuron will have the receptors for them. And they are neuromodulatory, so they're not going to change, or they're not going to cause any type of potential in the presynaptic cell. They're just going to change the way that cell is releasing neurotransmitter in the future. So what you notice here is that we've got 4 categories of neurotransmitters, and 3 of them are mostly neuromodulatory or always neuromodulatory. Only 1 category is neurotransmitters that are mostly not neuromodulatory, that mostly just cause local potentials. So most neurotransmitters are actually neuromodulators that are more involved with causing neuromodulation than they are with causing local potentials. Although a lot of those neuromodulators can also cause local potentials under certain circumstances, or some of the time. So we'll kind of go through the types of neurotransmitters individually, starting with the classical small molecule neurotransmitters. There are only three of them: glutamate, GABA, and glycine. Glutamate is always an excitatory transmitter, which means that it always causes depolarizing local potentials. It's also the most common neurotransmitter to be found in the central nervous system. So that means that out of all the neurons in the brain and the spinal cord, almost all of them, like most of them, can actually respond to glutamate. So they have receptors for glutamate, and lots and lots and lots and lots of the neurons that are in the brain and the spinal cord will release glutamate as one of their neurotransmitters. Glutamate is actually the #1 excitatory neurotransmitter in the central nervous system, which means that anytime a cell or a neuron is trying to excite another neuron or cause action potentials to occur in that other neuron, it's probably going to use glutamate to do it. Most depolarizing local potentials are just caused by glutamate. There's a variety of receptors for glutamate. One of the most important ones is the NMDA receptor, which you might have heard of, maybe not, but maybe you have. But the NMDA receptor is just really closely associated with learning and memory, and it's just really well studied, and it underlies one of the main models for how learning occurs that we have. So I won't go into the details of that model because I guess we probably just don't have the time, but the NMDA receptor is known to be very important for learning and forming memories. So that's glutamate. Then you have GABA and glycine, which are both inhibitory, which means they will always cause hyperpolarizing local potentials that take the cell further away from firing an action potential. GABA is more common to find in the brain, and glycine is more common in the spinal cord. So in the brain, the most common neurotransmitter is glutamate, which is excitatory, and the second most common is GABA, which is inhibitory. In the spinal cord, the most common neurotransmitter is still glutamate, and the second most common is glycine, which is inhibitory. So that means that when GABA, or when you have a neuron that is using GABA as its neurotransmitter, when that neuron has action potentials along its axon, it's going to release GABA onto the postsynaptic neuron, and that postsynaptic neuron will be inhibited, which means it's less likely to fire an action potential in the future, in the immediate future, as long as there's GABA signaling going on. And that actually ends up being really, really important. Just having that inhibition is actually equally important as excitation for actually processing information. If you only have excitation and you don't have any inhibition, you're not actually going to be processing information. That's just going to be basically random activity in the nervous system. That's not really related to anything specific and isn't getting any type of actual job done. You cannot process information without the inhibition. And then here I have an image of one of the GABA receptors. So this is a ligand-gated ion channel for chloride, actually. So when GABA binds to the receptor, it will open and chloride will flow into the cell, which hyperpolarizes the cell and inhibits it. You actually have a lot of different drugs that interact with the GABA receptor as well. So these three little spots here are places for different drugs to bind. The top spot is for benzodiazepines, which would be drugs like Xanax or what's the other one? Valium. Yeah, so like Xanax or Valium would bind right here and activate this receptor, opening it. Down here, flumazenil is actually kind of an antagonist. It's an antagonist for the receptor. So this is a drug that would be given to someone if they're kind of overdosing on Xanax or some related drug. You can give them flumazenil and it will bind right here to the GABA receptor and close it so that you don't have any more inhibition. And then down here you have the binding site for zolpidem, which is, it's Ambien. So this is sleep medication. Actually, in the further in or deeper into the receptor, you have the binding sites for barbiturates, which are kind of an older class of sedative that was used. It's not really used anymore. Very rarely, actually. Well, only phenobarbital is still used. Anyways, You have a number of different drugs that interact with the GABA receptor in different ways, either sedatives that are activating that receptor or drugs that are inhibiting the receptor, so closing that channel, preventing GABA-induced inhibition, and they would usually be used for reversing an overdose. The next type of neurotransmitter that we'll talk about is the neuromodulatory classical small molecule neurotransmitters. There's actually 5 of them: serotonin, dopamine, norepinephrine, acetylcholine, and histamine. So with the other classical neurotransmitters, with glutamate, GABA, and glycine, you'll basically find them all over the place. Like all over in the brain, you have neurons that make glutamate or GABA, and all over in the spinal cord, you have neurons that make glutamate or glycine. It's not that way for the neuromodulatory classical neurotransmitters. You actually don't find them all over the nervous system. The cells that make them are only found in very limited parts of the nervous system. You'll find them in like several small groupings, usually in the brainstem, or else, right by the brainstem. The brainstem being kind of the simplest part of your brain that's at the base of the brain. So the actual neuron cell bodies that make these neuromodulatory classical neurotransmitters, those are just found in those small little groups. But the axons for those neurons are going to actually leave those little groups and go all over the central nervous system. So they're going to branch a lot of times and end up putting or sending signals to basically most of the brain and spinal cord. Well, I should say especially the brain, so that means that most of the functions that are going on in the brain are affected by these neuromodulatory classical neurotransmitters. Most circuits in the brain are affected by these neuromodulatory classical neurotransmitters. So that means that you don't really have like specific functions that are associated with the different classical neurotransmitters. So each one of these 5 does a lot of different things, because basically every single function in the brain is going to be affected by them in some way, for the most part. So that said, there are like certain functions that each of these neurotransmitters has become kind of more prominently associated with. That doesn't mean that it's the main thing that neurotransmitter does at all. It's just the thing that we focus on most. So for serotonin, the main function it has that we focus on is mood. So, one of the common treatments for depression is the SSRI drug, which is the selective serotonin reuptake inhibitor. So that's going to block serotonin transporters and increase the amount of time that serotonin spends in the synaptic cleft. So it basically boosts serotonin signaling. And when you boost serotonin signaling in somebody's brain, a lot of people will become, happier or have a more stable mood. So it's a treatment for depression. It doesn't work for everybody, only for some people. And why it works, we don't know. We have no idea. But what we do know is that's only one of the many things that serotonin does. So for instance, psychedelic drugs like LSD also activate serotonin receptors, and those drugs make you hallucinate. So serotonin also has a role to play in your vision, not just in your mood, and actually would have a role to play not just in vision and mood, but in basically every major function that your brain does. For dopamine, same thing, it's associated with many different functions. Dopamine actually does have less functions than the other neuromodulatory classical neurotransmitters. It is a little bit more limited in its functional roles. Its main function is, well, it's kind of 2 main functions that it has. One of them is to help you do voluntary movements, and the second function would be related to reward, what they call reward, but basically addiction, wanting things and then becoming addicted to them. Dopamine has a big role to play there. So its role in voluntary movement is basically something we'll get into a bit more with chapter 12 when we talk about the basal ganglia. But the basal ganglia are basically gating your voluntary movement. So when you decide to do something, the basal ganglia have to kind of let that action occur for you to actually do it. and dopamine is necessary for that part of the brain to function correctly. So if you don't have that dopamine, you can try to do something, but you won't be able to do it. And that's kind of what's happening to people who have Parkinson's disease. Their dopamine neurons are dying, and so they don't have the dopamine, and that makes it harder and harder and harder for them to do simple tasks that they decide to do as they lose more and more of these dopamine neurons. Then the second role of dopamine is to promote basically desire, so wanting some stuff. And then when you have, I guess, inappropriate activation of dopamine, so you have like too much dopamine signaling, then that's when addiction can occur. So all addictive drugs actually increase dopamine signaling in the brain. Some of them do it directly, like cocaine directly increases dopamine release in the brain. But others will do it indirectly. So even drugs that don't affect dopamine directly will have those indirect effects that increase dopamine signaling in the brain. And that is the basis for addiction, actually. Then you have norepinephrine, which is most prominently associated with attention and just being kind of alert. In the central nervous system. It also has major roles to play in the peripheral nervous system. So the autonomic nervous system will use norepinephrine in a lot of different places to send messages, and we will talk about that more when we do our chapter on the autonomic nervous system, which we actually have two, chapters 13 and 14. Or I guess I should say we have two chapters on the peripheral nervous system. Only one of them is on the autonomic nervous system. Anyways, we'll talk more about epinephrine in chapters 13 and 14. But in the central nervous system, norepinephrine is mostly associated with being alert, being awake, paying attention to things. When you boost norepinephrine, it increases your concentration, helps with learning and memory, also has some undesirable effects as well. But those are the most prominent associations for norepinephrine. So these figures are showing you the distribution of cell bodies and axons that make dopamine, or four neurons that make dopamine, norepinephrine, and serotonin, and then the receptors for those transmitters. So here in this figure here, it's showing the distribution of the receptor types. So on the top you have dopamine. There's two types of dopamine receptors in the brain, D1 and D2. And so everywhere where you have blue, there's like a little bit of that receptor, not a lot, but there's a little bit. And then everywhere where there's like a different color, that means that there's either, well, green would be a medium amount and then red would be a lot. So you can see there is a little bit of dopamine receptors basically all over the whole brain. In the second brain image that's shown, it's kind of sliced, so you can see the internal structures, and that's just represented by little squares to show where the dopamine receptors are. So there's not a lot of parts of the brain that have a high amount of dopamine receptors, but basically the whole brain at least has a little bit. So the whole brain is going to be responding to dopamine in some way, essentially. For norepinephrine, you can see that there's a lot more of those kind of green and red areas. So norepinephrine receptors are very common to find in the brain, and they're basically all over the place. And it's the same thing for the serotonin receptors. These are actually only two types of serotonin receptors. Same thing for norepinephrine receptors. That's only two types. There's a lot of different types of receptors for these neurotransmitters. So they're basically all over the whole brain, is just the point. And then these more tan brains are showing the locations of the neurons that make those transmitters and where their actions go. So we'll start with dopamine here. The kind of purple blobs are the cell groups that make dopamine. So all the neurons that make dopamine in the brain will be located in one of these two groups, and then their axons are represented by the purple areas. So they're going out to most of the brain. For serotonin, the red blobs are showing the location of the cell bodies that manufacture serotonin, and then the arrows show where those axons travel, and you can see it's even more of the brain than gets dopamine input is getting serotonin input. And for norepinephrine, it's the same thing. You have the cells that make norepinephrine located in this green blob here, and the arrows show that the axons travel again, basically over the entire brain. And then you have histamine, which is most prominently associated with kind of the control of waking cycles or sleep cycles, and then control of hormones. So histamine is a little bit different from the other neuromodulatory classical small molecule neurotransmitters because its source is in the hypothalamus. And in the next chapter we're going to go through what the hypothalamus is and where it's at, but for now it's not part of the brainstem. So these classical or neuromodulatory classical transmitters, generally their source or the cells that produce them are found in the brainstem, but histamine is an exception because its source is the hypothalamus, which is not in the brainstem, but it's not too far away. But the hypothalamus is well, it does a lot of things, but one of the things it does, a couple of the things it does, is control kind of sleep cycles and control release of many of the hormones in your body. So histamine is associated with helping the hypothalamus accomplish those jobs. Histamine is also a very commonly used signaling molecule in the rest of the body by the immune system. So there's a lot of cells in your immune system that are able to produce and release histamine, and they will do that in response to any type of cell damage or infection, and it causes inflammation. If you produce kind of too much histamine, that can cause itching or allergies. So histamine is also associated with allergies, and that's why you have medications like Benadryl. Benadryl is a histamine receptor blocker. So some of those medications are able to cross the blood-brain barrier, and some of them are not. So if you're taking like a non-drowsy allergy medication, that is something that is not able to cross the blood-brain barrier, so it only affects the rest of your body and acts to control your allergies. If you're taking a different type of antihistamine, like for instance Benadryl, it actually does cross the blood-brain barrier. So when you take Benadryl, some of that drug is reducing your allergies or your inflammation, and some of it is actually crossing into your brain and affecting histamine receptors in the brain, and that's why it causes you to become drowsy. And actually, if you take way too much of it, it can even make you hallucinate. Then the last neuromodulatory classical small molecule transmitter is acetylcholine, which is mostly associated with attention and alertness, similar to norepinephrine is in the central nervous system. So also similar to norepinephrine, acetylcholine has a different role in the peripheral nervous system, which we will go over when we get into that those chapters. Acetylcholine is also unusual amongst the classical neuromodulatory transmitters because it's not actually recycled through a neurotransmitter transporter. Instead, you have an enzyme that degrades it, which is acetylcholinesterase. That's abbreviated ACHE. So when acetylcholine gets released into the synaptic cleft, it will bind to its receptor, and then instead of being pumped back into the presynaptic cell by a neurotransmitter transporter, it'll just be degraded by acetylcholinesterase into its component parts. And then those component parts would actually be pumped back into the presynaptic cell and kind of reassembled to form acetylcholine again. And I guess the final note I'll make about these neuromodulatory classical transmitters is just about their chemistry. So serotonin, dopamine, norepinephrine, and histamine are all classed as monoamines. That is kind of the type of chemical that they are. When we say that they are monoamines, we mean that they have a ring and an amine group. So here you have images of serotonin, dopamine, and norepinephrine. You can see they're all very closely related to each other structurally. But here's the ring that they all have. And then the amine group is a nitrogen with hydrogens attached to it. All of those monoamines, all the monoamine transmitters, are degraded by the enzyme monoamine oxidase. So after they get released at the synapse, they bind to the receptors. Then when they let go to recycle them, they're going to be pumped back into the presynaptic cell by a neurotransmitter transporter, and then once they get back into the presynaptic cell, into that axon terminal, you have the enzyme monoamine oxidase, which will degrade them, and then they'll be recycled. And I'm only really mentioning that because There's a lot of medications that kind of interact with those transmitters, especially serotonin and norepinephrine. So a lot of medications for depression interact with serotonin or norepinephrine or both. And then medications for ADHD tend to interact with norepinephrine. So whenever you're on a drug like that, generally what it means is that the level of that neurotransmitter is being boosted in your system, in your brain, usually by blocking the transporter for it so it can't be recycled as efficiently. And you may be told to avoid eating certain things like grapefruit juice or St. John's wort, which is like an herbal supplement. The reason why you're told to avoid those things is because they contain what we call an MAOI, which it just stands for monoamine oxidase inhibitor. So St. John's wort and grapefruit juice have compounds that will actually inhibit the monoamine oxidase enzyme that degrades those transporters. So, degrades those transmitters. So if you're taking a drug that boosts the level of that transmitter in your brain, and at the same time you're taking another drug without even necessarily meaning to, that prevents that transmitter from being degraded, now you're going to get an even larger boost in the level of that transmitter in your brain. And that might end up actually being a bad thing for you. It is possible to have too much of these transmitters. In particular for serotonin, if you get too much, you can get a syndrome called serotonin storm, which can be fatal. So if you've ever been told to avoid grapefruit juice or St. John's wort with a medication that you're taking, that's the reason why. And that's based on the chemistry of those neurotransmitters. They're all degraded by the same enzyme due to their chemistry as monoamines. Then dopamine and norepinephrine are further kind of classified as catecholamines because their ring has 2 hydroxyl groups on it. The hydroxyl is just this oxygen with a hydrogen on it. And catechol actually is just a ring like this with 2 hydroxyls on it. So these are catecholamines because they have an amine group as well. And finally, I just have our images showing distribution of these neurotransmitters. So on the bottom here, you have the source of histamine, would be this red glob, and kind of in the middle of the brain, so that would be in the hypothalamus. And then you have arrows coming out from it showing the axons of those neurons and where they travel in the brain. So you can see is basically the whole brain and down into the spinal cord as well. Same thing for acetylcholine here. Its main source is in the brainstem, and it also has a couple more sources in the hypothalamus. And its axons will also travel basically around the whole brain. And then up here in the upper left, we have just the receptor distribution for acetylcholine. So whether it's pink, blue, or green, those are just kind of color coding for different types of acetylcholine receptors. So you can see that those are basically all over the whole brain and throughout the spinal cord. I wasn't able to find a comparable image for histamine, actually, but its receptors are similarly widely distributed. So I've been mentioning a few times now that these small molecule neuromodulatory transmitters, your serotonin, dopamine, norepinephrine, acetylcholine, histamine, are kind of being produced in like small or by small groups of cells that are usually found in the brainstem or also nearby the brainstem, but then the axons for those cells kind of leave that location and start traveling and branching all over the brain. And so that basically the whole brain is receiving input using these neurotransmitters, is receiving input from axons that signal using these neurotransmitters. So that means that these transmitters have their fingers into everything, or into all basic processes in the brain. And that is true, but it's not the case that literally every single neuron in the brain is being contacted by axons that release these neurotransmitters. And it's also not the case that every single small structure within the brain is being contacted by axons that release these neurotransmitters. So the brain is really, really, really complicated in terms of its structure. We're going to go through a little bit of that in the next chapter, but we're really barely going to be scratching the surface, to be honest with you. We're going to go through kind of the main parts of the brain and then a couple of their really important subparts. But if you're really trying to learn the anatomy of the brain, you're going to zoom in on those subparts and realize that they got like they're full of structures that are smaller inside of them, and like the structures got substructures, and even some of the substructures got their own substructures. It's crazy complicated. There's hundreds and hundreds of different structures in the brain. Especially if you're counting the substructures as well. So not literally every one of those structures and substructures is going to get input from serotonin neurons or dopamine neurons or norepinephrine neurons, but all major circuits will. And the image here is just kind of showing a little bit of a more detailed view of serotonin axons and where they travel in the brain with some of these substructures in the brain. And honestly, it is still quite a limited picture because it gets crazy detailed. But as you can see, here's a couple making kind of a triangle shape, triangle pointing left, and the tip of the triangle is receiving input from serotonin axons, but the base of the triangle is not. So there's a substructure that is not getting serotonin axons, and serotonin will not be directly involved in the function of that particular substructure, although it very likely is involved in the function of the overall circuit that that substructure is functioning as a part of. Okay, so now on to the neuropeptides. A peptide is defined as a protein basically that's small. So that's not the literal definition actually, but you know, it's a chain of amino acids that is 50 or less amino acids long. And then if you're over 50 amino acids long and you're folded up, then you are a protein. So there's a lot of peptides in the body, and they have a lot of different functions. It's common to find them acting as signaling molecules or as serving different roles, various roles. And in the brain, you have a lot of peptides that act as neurotransmitters, so we call them the neuropeptides. There's a bunch of them. There's like over 100 of them. Most of them are neuromodulators, so they're usually going to be acting not by directly causing local potentials, but instead by just kind of altering the function of the synapse, either changing how the presynaptic cell releases neurotransmitter or changing how the postsynaptic cell responds to that neurotransmitter. And they would accomplish those goals by either changing protein function or gene expression in the cells that are receiving signaling by these neuropeptides. A lot of the time, you'll have an axon that actually signals using multiple neurotransmitters, and one of them is one of our classical small molecule neurotransmitters, and the other one is a neuropeptide. So that means that from one axon terminal or at one synapse, you would have not just that classical small molecule neurotransmitter released, but you could also have the neuropeptide released as well. So that's two different neurotransmitters that are probably going to do different things in that postsynaptic cell because they're going to have different receptors. You could even have multiple neuropeptides being released from the same synapse at a time. It just kind of depends on the synapse. In general, whenever you have that type of situation, when you have action potentials coming in to the axon terminal, they open those voltage-gated calcium channels and you get some calcium entering the cell, and that's going to cause neurotransmitter-containing vesicles called synaptic vesicles to fuse with the membrane of the axon terminal and release that transmitter. So in general, you're going to have if you have just like a couple of action potentials coming, then you're going to have a few of those voltage-gated calcium channels opening, and you're going to get some or a little bit of calcium entering the axon terminal, and then that's going to trigger the release of the classical small molecule neurotransmitter. Then if you get like a bunch of action potentials traveling along that axon and arriving at that axon terminal, you're going to open a bunch of voltage-gated calcium channels. You're going to get a bunch of calcium entering the cell, and at that point you will also have the neuropeptide transmitter be triggered to release into that synapse. And then the last thing about neuropeptides that kind of makes them even more complicated is this thing called volume transmission. So we've talked about neurotransmitters being released at synapses, but for neuropeptides, sometimes they're not actually released at a synapse. Sometimes they're released along an axon. So sometimes as an axon is traveling through the brain, before you get to the end of that axon, you're going to have kind of like a bulge in it, or a place where it kind of swells up. And at that place, which we would call it a varicosity actually, similar to a varicosity in a vein as you get older, at that little bulge, you would actually have neuropeptides that are ready to be released. So as an action potential is traveling along that axon, it's going to pass through these varicosities, and when it gets to them, when it passes through them, the neuropeptides there will be released. And they're not released into a synapse or anything like that, they're just kind of released into the extracellular matrix of the brain, and they're going to start diffusing through that extracellular matrix, and they may encounter multiple synapses as they diffuse outward and activate receptors on those synapses. So here you have kind of an image showing volume transmission. In red you have an axon traveling through the brain, and you can see it's branching to form two different branches, and you've got these little bulges on it in a couple of places. So those are the varicosities. So if you have an action potential traveling along this axon, it's going to just pass on by, and when it passes by these varicosities, it's going to cause them to release their neuropeptide transmitter. That neuropeptide is going to start diffusing outward through the tissue, and these kind of red clouds around the varicosities are showing where that neuropeptide would be able to diffuse to. How far it diffuses in the brain is going to be kind of limited by the location of enzymes that degrade it. So right here where the kind of red cloud is ending, that's where you're going to find a lot of enzymes that are able to degrade this neuropeptide so it stops traveling further. And if you zoom in on one of these areas where the neuropeptide is diffusing, you can see it might look something like this. So here you have your varicosity, the little red dots of the neuropeptide being released. They're diffusing through the extracellular matrix of the brain. And they're encountering here a synapse. And you have some receptors for them on that synapse. So they're going to do something to that synapse, probably changing protein function or gene expression in one of those cells and altering the way that it signals in the future. Oh, and by the way, these blue cells, those are astrocytes. So this, volume transmission and multiple neurotransmitters being released from one synapse, like that stuff's not going to be on the test or anything like that. I just wanted to kind of mention it to you to increase your awareness of the fact that the brain is really complicated. It's like crazy complicated. And this is really the reason why we don't understand like how it works. It is too complicated. But maybe one day. So I'll just go through a few examples of neuropeptides before we move on to the retrograde neurotransmitters. The first example I have is opioids. So opioids are all neuropeptides. You can kind of also call them endorphins. So whenever your body is kind of naturally suppressing painful sensations so that you don't become aware of them, it's actually releasing these opioid neuropeptides in the brain. And their primary role is just to suppress your perception of pain. There's actually a number of them. It's not just one, so that's why we call them the opioids. And all of our narcotic painkillers, like morphine, oxycodone, like Vicodin, all those things, also heroin as well, all opiate drugs, they will all interact with opioid receptors. So they're all kind of binding to the receptors that are intended for these opioid neuropeptides, and they're activating those receptors. So when you're taking those drugs, you're actually increasing the amount of pain suppression in your brain, as well as doing other things. They have other roles as well. They can actually affect appetite also. Yeah, so it's not just pain suppression, but that's kind of like the main thing that we think about them in connection with. Then you have substance P, which is actually the first neuropeptide to be discovered, which happened back in the 1980s. So we haven't even known about these neuropeptides for all that long. And it was actually discovered in the peripheral nervous system, where you have sensory neurons for pain that are called nociceptive sensory neurons signaling with substance P. And the final neuropeptide that I'll mention is oxytocin, which you might have also heard of. It's kind of starting to drift into the popular awareness, and it's known as a neurotransmitter that promotes like bonding, social bonding. People like empathizing with each other, trusting each other, and also maternal behavior, so like kind of parenting type behaviors. So those are some of the things that oxytocin does. It also does other things, and it actually maybe have some kind of dark functions as well. So we think of it as being kind of this happy, feel-good molecule that helps you like, bond with the people that you're close with and have, close, loving relationships. But actually, there have been experiments done where you kind of hook a person up to like an MRI, and when you can show them images of somebody that they consider to be not part of their social group kind of getting hurt, like maybe falling down or something. And when you're measuring their brain activity when they're viewing this image, you can kind of pick up a response in kind of pleasure areas of the brain. And if you've given them an injection of oxytocin beforehand, the pleasure response actually gets stronger. So people will enjoy watching someone that they don't consider to be part of their group get hurt more if they have more oxytocin in their brain. So it's not just a good thing, it could be, sometimes in certain contexts, like not a great molecule actually. And it's also a hormone in the rest of your body, so it's released in childbirth to stimulate contractions of the uterus, and it's released during breastfeeding to stimulate milk ejection. In the brain, there's not a lot of cells that release oxytocin. The ones that do can actually connect to each other using these synapses called, well, not synapses actually, but junctions. Cell junctions called gap junctions. That actually talked about in chapter 4 with histology. And when you have cells that are connected using these gap junctions, I have an image of it up here. Basically, it's just a couple of pores on either side, or in each cell's membrane that kind of hook up to each other, and then ions can go across. So if you have an action potential arriving in this axon terminal, that means you're going to have sodium entering this axon terminal. The sodium ions can just cross into the next cell through these gap junctions. And that means that the action potential can basically just continue on in the next cell, or you can have a local potential starting directly in the next cell, depending on what type of ion channels you have in that cell. If you had voltage-gated channels, it would probably start an action potential. Otherwise, it will just be a local potential. So whenever you have neurons that are connected by gap junctions like that, we call that an electrical synapse, because you're not sending the message from one neuron to the next using chemicals. You're just using ions, which I guess are chemicals technically, but when they're moving, they're carrying a current with them. So you have actually a current flowing from one cell into the next cell. So we call it an electrical synapse. So your neurons that release oxytocin can actually hook up to each other using those gap junctions to form electrical synapses. That way, when one of them starts firing, all the other ones will start firing at the same time as well. The action potential will just kind of spread to all of them, and then they're all going to kind of release oxytocin at one time, and that gives you a big burst. And you need that big burst of oxytocin in order to do certain things, like actually stimulate milk ejection. So here I have just an image that's kind of showing how multiple neurotransmitters can be released from the same synapse, or from the same presynaptic neuron at a synapse. So here we have our presynaptic neuron and its axon and axon terminal. Then we have the postsynaptic neuron on the other side. And this presynaptic neuron has 3 different neurotransmitters that it's releasing. Well, actually 4. These little circles are its synaptic vesicles that are storing neurotransmitters to be released. The blue triangles are a small, just a regular classical small molecule neurotransmitter, so probably either glutamate or GABA if this is in the brain. The red dots are the neuromodulatory classical small molecule neurotransmitter, so maybe dopamine or serotonin. And then the big green dots are a neuropeptide transmitter. So when you have action potentials coming down this axon at kind of a steady rate or kind of a slower pace, you're going to have some amount of calcium entering this axon terminal. And that's going to trigger these regular kind of small molecule transmitters to be released. So your glutamate, maybe it's going to be released with kind of just regular slow stimulation. I know it looks a little fast, but that's slow for the brain. Then on the top, we have like bursts of action potentials, like really thick, dense bursts that are coming through. So the axon would be firing action potentials really, really quickly, basically as fast as it possibly can in this case. And when that happens, you're going to get a lot of calcium entering this axon terminal, and then some of these bigger vesicles that have the neuropeptide in them, those are going to start fusing with the axon terminal membrane as well and releasing their neurotransmitter. Then these squiggles are receptors, G-protein coupled receptors for that neuropeptide. These are your ligand-gated ion channel receptors for the small molecule transmitter like glutamate. And then over here, you actually have nitric oxide being released as well, which is another transmitter, which we'll talk about right now. Okay, with our retrograde neurotransmitters. A retrograde neurotransmitter, again, is just a transmitter that goes backwards. So it's released from the postsynaptic neuron and it goes to the presynaptic neuron in general. You have two types of retrograde neurotransmitters, nitric oxide and the endocannabinoids. Nitric oxide is unique amongst neurotransmitters because it is a gas. And endocannabinoids are also unique because they're the only lipophilic neurotransmitters. So they're actually produced by kind of snapping off little pieces of the lipids that cells have in their membranes. So in general, how these retrograde transmitters work would be that you kind of have like normal activity at the synapse. So you have your action potentials coming down into the presynaptic neurons, axon terminal. Voltage-gated calcium channels open, calcium enters the axon terminal, it binds with proteins on the synaptic vesicles and activates those vesicles so that they will kind of go to the membrane, they'll migrate to the axon terminal membrane and fuse with it, which releases those neurotransmitters. The neurotransmitters are going to cross the synaptic cleft and bind to the receptors on the postsynaptic cell, and something will happen, maybe it's a local potential. So at that point, that's when the retrograde neurotransmitter would come into play. When your regular neurotransmitter has bound to its receptor, that is actually going to trigger the release of the retrograde neurotransmitter, and it will go backwards across the synaptic cleft back to the presynaptic neuron and bind to its receptors on that neuron. So these are always neuromodulatory. They're never going to cause a local potential. They will always just alter the way that the synapse is functioning by changing the way that that presynaptic neuron is releasing neurotransmitter in the future. And because you're always kind of releasing them in response to activity at the synapse, they end up usually being involved in negative feedback loops. They're always going to be involved in some type of feedback loop, but it's usually going to be negative feedback, which means that your forward transmitter or your regular transmitter binds to the postsynaptic neuron. The postsynaptic neuron then releases its retrograde neurotransmitter. That neurotransmitter binds to the presynaptic neuron and causes it to reduce the amount of forward neurotransmitter that it releases. So in the future, when you have action potentials come down this axon, you're going to release a smaller amount of the forward neurotransmitter or the regular neurotransmitter than you usually would. So if our regular neurotransmitter is glutamate, then we have glutamate, crossing the synaptic cleft, stimulating the postsynaptic neuron with depolarizing local potentials. Then the postsynaptic neuron releases its retrograde transmitters. Those go backwards across the synaptic cleft, and they cause the presynaptic neuron to release less glutamate in the future, which means that now, in the future, when you have stimulation at that synapse, you're going to have less glutamate released, you're going to get smaller depolarizing local potentials in the postsynaptic cell, and overall that cell will be kind of like inhibited. It's going to be less likely to fire an action potential because it's having smaller local potentials now, smaller depolarizing local potentials. On the other hand, if we're talking about a presynaptic cell that's releasing GABA as its kind of regular neurotransmitter, then we're going to have the opposite effect. So GABA would be released, crosses the synaptic cleft, binds to its receptors on the postsynaptic cell, and causes hyperpolarizing local potentials that are inhibitory and actually prevent that cell or make it less likely for that cell to fire an action potential. Then that postsynaptic cell is going to release its retrograde neurotransmitter, which will go backwards across the synaptic cleft and bind to its receptors on the presynaptic cell. And then in the future, the presynaptic cell will release less GABA when action potentials arrive at that synapse. So in the future, when you have your action potentials come in, you're going to release less GABA, which means that the local potentials you get in the postsynaptic cell are going to be smaller. But these are hyperpolarizing potentials, so when they're smaller, you have less hyperpolarization, which means less inhibition, which means that neuron is now more likely to fire an action potential. And so I'll just kind of go over briefly how nitric oxide works. This is not going to be on the test or anything like that, but just, to give you kind of an illustration. And nitric oxide is really unusual for neurotransmitters because it's a gas. Since it's a gas, you really can't control it very well. It's really, really small. And it's also nonpolar, which means it will get through a cell membrane very easily. There's no way to control like where nitric oxide is going to go within the tissue once you produce it. So that means that it's not just going to go actually backwards across the synapse, it's just going to be kind of spreading out from the point at which it's produced. So it'll end up impacting the presynaptic cell and also any other cell in the nearby area that has receptors for it. So, for instance, maybe there's an astrocyte nearby that might have receptors for it. Maybe even other neurons, maybe even the neuron that produced it. So there's no way for the brain to really make sure that one particular cell is being affected by nitric oxide, apart from just making sure that other cells don't have the receptor for it. But probably most cells kind of do. Since it's able to diffuse through the membrane very easily, it doesn't need a receptor that's located on the membrane. Instead, its receptor is going to be located inside of the cell, and it's actually an enzyme called soluble guanylyl cyclase. So I have here a diagram kind of showing how nitric oxide works. So here we have our presynaptic cell. This cell releases glutamate as its primary neurotransmitter. So here it has a vesicle filled with a synaptic vesicle filled with the glutamate. When you have action potentials come down this axon to the axon terminal, voltage-gated calcium channels will open. Calcium is going to enter the cell. That's going to cause the synaptic vessel to fuse with the cell membrane and release the glutamate. Glutamate is going to cross the synaptic cleft and bind to its receptor on the postsynaptic cell. That receptor actually might be a G-protein coupled receptor. There are G-protein coupled receptors for glutamate, but in this diagram it's shown as the NMDA receptor, which is a ligand-gated ion channel receptor, but it's kind of a special one because it's also involved in learning and memory. So when glutamate binds to this receptor, it's going to open it. In this case, this particular ion channel allows calcium to enter the cell. So now you're going to have calcium entering the postsynaptic cell, and that's actually a signaling molecule in the cell. What calcium is going to do in this place, or in this case, is turn on this enzyme, neuronal nitric oxide synthase. And this enzyme is going to manufacture nitric oxide from arginine. So ultimately, you're going to get nitric oxide here produced. That nitric oxide is going to start diffusing out. It actually encounters its receptor in this cell that produced it, soluble guanylyl cyclase. And it's also able to diffuse out through the tissue to other places. So it's diffusing here into the postsynaptic cell. It's also diffusing into a nearby astrocyte. It's also diffusing into another cell, another neuron that is actually sending its axon nearby. So nitric oxide is going to get into all of these cells and locate its receptor and activate that receptor, the soluble guanylyl cyclase. When that receptor activates, because it's an enzyme, it's going to start catalyzing a reaction. Actually, what this enzyme does is it makes cGMP, which is also called cyclic GMP. So it's producing cyclic GMP in all of these cells. And then, cyclic GMP is actually a signaling molecule in the cell, so it's going to go and, carry that message and do some stuff. Activate some enzymes, turn some enzymes off. What it does exactly is actually going to depend on the cell and what enzymes are there that are sensitive to cyclic GMP. But in the presynaptic cell, the ultimate effect is probably going to be to reduce the amount of glutamate that is released in the future. So we're releasing a good bit of glutamate molecules here in response to an action potential. Maybe in the future we'll release just a couple in response to an action potential. But it can also do other things in the cell as well. It can change gene expression as well. And overall, nitric oxide is important for learning and memory, which makes sense since it is a neuromodulator. But you actually have to have it for learning and memory. Same thing for the endocannabinoids, actually. Those neurotransmitters, or I guess I should say the receptors for the endocannabinoid transmitters, are actually the receptors that are activated by marijuana, and by the other, not the endocannabinoids, but the cannabinoids. The endocannabinoids are the cannabinoids from inside your body. The other cannabinoids are from outside your body. So all those cannabinoids are actually going to just be activating receptors that are intended for the endocannabinoids. You actually have to have that for learning and memory to occur. If you block those receptors in experimental animals, they're no longer able to learn or to forget. So finally, I just have kind of a summary slide that's kind of wrapping up what we just went over with the neurotransmitters. So we have our four groups of neurotransmitters: the classical small molecule neurotransmitters, the classical or the neuromodulatory classical small molecule neurotransmitters, the neuropeptides, and the retrograde neurotransmitters. So our basic synaptic transmission that we kind of talked about first, the basic model, is always involving those classical small molecule transmitters, which are glutamate, GABA, and glycine. Glutamate means on, GABA and glycine mean off, so glutamate causes depolarizing local potentials, GABA and glycine cause hyperpolarizing local potentials. GABA is more common in the brain, glycine more common in the spinal cord, and glutamate is the most common neurotransmitter everywhere in the central nervous system. So when you have synapses that are releasing glutamate, GABA, or glycine, basically that neurotransmitter is going to bind to a ligand-gated ion channel receptor and open it, and then you're going to have ions crossing the membrane, and that causes your local potential to occur. So that's your basic neurotransmission. It's either promoting an action potential in the postsynaptic cell if it's glutamate, or it is reducing the likelihood of an action potential occurring in the postsynaptic cell if it is GABA or glycine. Then on top of that, layered on top of that, you have neuromodulation, which ends up being really, really important, and most neurotransmitters are involved primarily with neuromodulation. So in neuromodulation, you are changing something about how the synapse functions. Either you are changing the way that the presynaptic cell releases neurotransmitter, or you are changing the way that the postsynaptic cell responds to neurotransmitter. In either case, you can get that job done by changing protein function or gene expression in either one of those neurons. And this neuromodulation is essential for learning things, for forming memories, also for forgetting things. It can, in extreme cases, not just change the function of the synapse, but if you have a lot of kind of new genes turning on, new proteins being made, you can actually build new synapses for learning and memory. So the neurotransmitters that get that done fall into our remaining three categories. First, you have the neuromodulatory classical small molecule transmitters, which are dopamine, serotonin, norepinephrine, histamine, and acetylcholine. Then you have the neuropeptide transmitters, which are all peptides, so small chains of amino acids. Those neuropeptides are kind of funky because you can actually release them along with a classical neurotransmitter, like from the same synapse as the classical neurotransmitter. You can also release them at other points along an axon, in which case they would just start diffusing out through the extracellular matrix until they encounter receptors, and that's called volume transmission. And then finally, you have the retrograde neurotransmitters, which are also funky because they go backwards. And there's only two types: the nitric oxide gas and endocannabinoids, which are they're not lipids themselves, but they come from lipids, and they are lipophilic or hydrophobic. So the retrograde transmitters go backwards from the postsynaptic cell to the presynaptic cell, and usually reduce the amount of neurotransmitter that presynaptic cell releases in the future, which causes a negative feedback loop.